plant. Like that, I mean, you can see thousands here. Now we have, say, 17, 18,000 plants are in India also. By documented and I mean, already identified by Mortals in India also. So we have a large richness about its uses of traditional knowledge about it. This is one example of Cervical also, how we can do it in the past, how people are using this plant, how it can develop one by one, year to year, by their practices and their knowledge also. Same time, Hippocrates also in Europe, they are also working the same line. There's no further medicine is okay, next one. So these are some examples why it's the same way diasporics also. In Europe also they identify these plants and not developed Greek medicine also. So it was seven human is there. Naturally human go to the forest, how to use it for the fire, for food, for medicine. So suddenly evolution was going on. So the Europe part, the Indian part, like India, we have tradition like Buravia. You must have seen in village also. So they have a very common one. People use it. I mean, Ayurveda is it here, but if you go to villages, they just have a root of that, and like we have fluid nowadays, people are using that. So, Buravia, it is a very protective directly. So, we have done the experiment. I think 3 years it took for us to extend the root, whether it is small root, big root, which season we have to collect. So, that work we have done it in the, with the collaboration CDRI Lucknow. Took 3 years. Finally, we said it is a June month, and this size of root with hydroalcohol extract is active. So, this site is a validation of claim. That was also back perception. If we get some identification from extract, if we identify some molecule, so we can go for the patenting also. Next, how to do it, I will come later on. So, this was an example of that. Next one. Similarly, for traditional user, Texas soil, well known the Ayurveda also, European plant also, India also it has. Later on we got the taxon from this for the agricultural compound. This is a medicine. This is also from leaves from the traditional. Next one, similarly for aconites. There is no it's being used, it's still being used in Himalaya also for fever and other things. But after doing the research, CDI also, with the I.S.G.B.R.D college also, it's an anti-inflammatory activity. So like that, people are working for last like 50 years, 50 years, then you get some medicine, you do some activity, but to save drug, there are a number of steps. I will tell you what are the steps you have to follow to save drug. Next one. Like people are a very common plant in Himalaya also, up to 8,000 above, you get this plant, very beautiful, and very bitter also. Now in CDRI, we are going to have a first phytopharmaceutical drug on the Picoriza, it's called Picrolid, and it is good for your liver also, enlarged liver also. This is going to be one of the best drug for the CDI for India. Next one. So similarly, there are a number of examples of that. Now what we have, I just show you, we have rich biodiversity in India. Next one is we have the click, and we have traditional based information, indigenous based technology, we have so much medicine, we know folklore information, and we have the modern tools now. After you also have a pharmacy department, you have the pharmacist also, pharmacy, CDI, number of institutions of a government, they have expertise in the governing drug also. What we have to do is, we have to convert all these three into a product, we can say our drug, and then a phytopharmaceutical molecules. This is third category of drug, is newly categorized, and we can go on nutraceutical also, and the pharmaceutical also. So this way we can convert our biodiversity, taking this knowledge, and we can make product which can help our healthcare needs and also develop economic wealth and the employment generation to young people. This is what we have to do it. We are doing also. No, hello, please listen. Otherwise, you can go outside.
Can you be silent, please? You can focus on the lecture so you can ask the queries.
Disturb the floor. Otherwise, you can discuss the thing outside. So, next one. So, what we are saying, how we can do prospecting of plant, you can go for I.S.G.B.R.D, Ayurveda, you can go, but that way, whatever plant material you have, you can make the extract powder or you can extract water the hydroalcohol. That's all, only this. You cannot not go beyond that. If you go beyond that, now there is another category of a solvent. You can have it, butanol, ethyl acetate, other fraction also. With that fraction, we can active fraction can go. We call it pharmaceutical drug. This is a new category. Only 2016, we got the permission from the Drug Control Act from Government India. It has benefit. What benefit I will tell you. And lastly, modern medicine like you have the paracetamol, antibiotic, single molecule is here. So this is the in between the new category of drug that is good for India. Although we are late by the more than 10-20 years, China and Europe countries they are having that. But this category has plus point because it has in between you have to do everything which are defined to prove its efficacy, safety, clinical trial as for the modern medicine. But same time it is not necessary to have a single molecule. You can have hundreds of thousands of compounds, but extract fraction should be active. We have to safety, efficacy, and clinical trial in this case also. So it is in between one, and this can be prescribed by the modern medicine doctors also and allopathic also. While in ASU that only Ayurvedic doctor or Unani doctor, Siddha doctor, they have right to prescribe the drug also. And modern means all everyday what they are prescribed. So, but time is the duration. If say ASU that Ayurvedic medicine, you can make it 4 to 5. Yes, means the combination you can make it by standardization, quality control, doing a validation, as per the new drug, and it takes 50-30 lakhs. This is roughly estimation. Pharma it will take 5 to 8 years, maybe 10 years, and more than 2 crores are required for develop one phytosanitary. And for single, as you have seen, morning also, yesterday also, it looks millions of rupees are required for to develop identify one molecule, and which is very active, safe, so you can market it. But it has more than 100 crores required, more than 15-20 years are required. So it is very, to get the single molecule is a long-term approach. In India, we have the CDRI is working on that, and I think more than out of 13 drugs, modern medicine were developed by the CDRI. It is our sister labs, CSI labs is there. There are RT ether is there, and Google Lipid is there. There are RT ether, and there are number of rules. Saheli or the centromine is there. Number of compounds they have developed. So you can see, India only we have 3. You can market only 3 categories. Ayurveda you can market in India only. India or where Ayurveda is known, like Bhutan, Nepal, Bangladesh, Pakistan, but not beyond that. If you want to export your medicine in Ayurveda medicine, America, it will not allow. They will not allow. You can say it is nutraceutical, then they will allow it. This is the legal things which one should know. So that's one. So these are basic things for the student also. What we have, just I will tell you, these are traditional approach. We know the plant traditionally in our Ayurveda or folk medicine. We collect it, then after collection, do the extraction, after drying, make the fractionation, and go for the activity guided test for that. So you can go the homology activity of that, whatever you want, and do the animal study, in vitro and in vivo also, and directly if it is safe, you have to do safety as per the Drug Control Act. If it is safe, then you can make the encapsulation. You can make doses form, and directly you can do the quality control, and after doing clinical trial, phase 3, you can go to directly you can go to market. This is a way of things, how to make the perspective plans for the drugs. Next one. So in short, this is the molecule, mice, and man. If I enter plants, a compound is there. What you want to develop drug, you have to do chemistry, you isolate this, test in animals, and then clinical trial required. This is the modern molecule, mice, and man. But why? What we have in Ayurveda, we have knowledge of traditional medicine. Directly you will prove. Suppose plant and medicine, Ayurveda, you prove. If it is good for legislative, you prove the legislative model you have, and directly you can do the clinical trial, desirable, not compulsory. Unless it is new indication. Then in fact, we need the clinic. We call it liver pharmacology. That is the best way for our India to get, because we have a large knowledge information. You can have a platform to work on that, and your student can do it, and directly you can do the clinical. This is the way we can do it, and this is the different country, USA, Japan, China, and you have to see the pharmacopoeia of that country, and accordingly you have to make your formation. What are required on that? In Japan, China is different. In France, we are the all pharmacopoeia legal document which are required to market and drug to these countries. Every country has their own pharmacopoeia. Like India, we have a drug considered Act 1940. So everything is mentioned there, how to do it, who can market, what compound, what are the requirements to say a drug, Ayurvedic drug, or United drug, or phytopharma, everything is there. So you have to learn it. Particularly for the pharma student, they know what they just course also how to do the drug development. So particularly for the student for the pharma, it is very, they know the things also. So this is a way for the, if you have interest. So it is not like that you have a drug here, you can directly export to China or you can export to France. We have to see what are the permission we have to take. Permission, they say these are requirements. So we have to take out. So next one, so I will quickly go through it, how to make the Ayurvedic medicine or herbal medicine. What is different with Ayurvedic medicine and herbal medicine? Ayurvedic medicine are those herbal medicine which are mentioned in authoritative books of Ayurveda, like money plant, stevia, money plant, mentioned Ayurvedic textbook, authoritative books. Then you can say Ayurvedic medicine. So there are more than 80 books which are recognized by the government. Ayurvedic medicine, means to Ayurveda. These are the books, the plant which are mentioned in this book, we will call it Ayurvedic medicine or Unani medicine. If you call, there are books in Ayurveda also, Unani also, Siddha also. Accordingly, there is a tax exemption also. There is a legal point. So how you can make it? Next one. So this is the act of that. Only thing, if a powder is there, you can make the aqueous. Aqueous means water extract, you can make it. After making water extract, you can decant it, dry it, then you can make it tablet. And what you require, safety not required, if you are mentioned in the, if you are making the formulation, in the same as per the Ayurvedic test. So already because you are tested, so no safety required. Only no pharmacological activity required. If you are say Brahmi, I am making and I will market it for the brain tonic, no requirement, right? But you say, no, not Brahmin for the brain, it is good for say skin, for the skin. Then you have to show pharmacological activity, not the safety. So we can go aqueous up to the hydroalcohol. This is the way, as per the Drug Cosmetic Act 1940, you can go up to hydroalcohol, you can use steps. Next one. So this is the how one can go, just summarize it like development of phyto medicine, phyto plant-based medicine. You select the plant, make the preparation. Preparation you can have water or hydroalcohol. Mix of alcohol 50%, 50-50, 40-60. It's up to you. Hydroalcoholic, if you gain the activity, you can do the extraction screening of that, and directly this process, if you find the activity, you can go for the IUS mode. You can use the IUS mode. If you are not satisfied, do the fractionation, sub-fractionation, okay, and do the every fraction, you have to do the pharmacal activity of that. These are activities you are getting. Then, if you identify this fraction, active fraction, you can go as a phytomedicine, a phytopharmaceutical in this case. Because later on, if you further do the fractionation, some activities is not there. Because now in Dr. earlier speaker also told you, it is a synergy, not a single molecule, it is a compound, there are a number of compounds are there which enhance the activity, work and catalyst. Synergy is there. If it is not there, if you do further fractionation, activity will be not there. So we have lost number of compounds this way. Now government will say, if it is not active, that fraction go back to the earlier fraction and do the activity, found the activity, safety, efficacy, and you can market it. Now this is for a phytopharmaceutical basis. And if you have to show safety, pharmacokinetic, genotoxicity, pathogenicity, whatever requirement of modern medicine is for safety point of view, you have to do it. So safety study is required to say phytopharmaceuticals. And if you can identify the active compound, one molecule, or you have to show the stability, what are the expiry date, or you can fix the expiry date also, stability, and later you can do clinical. Clinical are immersed in the phytopharmaceuticals. This is the status of in India, three categories in India that I recognize in India. I use medicine and phytopharmaceutical and IND, that is a modern medicine. Next one. So this is phytopharmaceutical. In some, just in short, you say it is a fraction, active fraction, which is from the derived from the plant, and you have to show safety, efficacy as per the modern medicine. So we call it phytopharmaceutical. Only 2015 it was recognized by the government, and later on now I think there in market I think number of drugs are there. So this is a new drugs of category which we can also a Ayurvedic doctor, even a modern medicine MBBS can also prescribe, and you can export this drug also. This is the plus one. Next one. So there are number of compounds. Modern medicine, now I come to modern medicine. Modern, some simple compounds. You have organelle, we have the oxo, xylene, anti-cancer also. Like I.S.G.B.R.D, we have chloroquine, which is anti-inflammatory also, anti-cancer also. That is identify the compound. The identification of compound is okay after 11. But to say drug, you need to do so many things. And require, I told, maybe 10 years, 5 to 10 years, 20 years. To say you are speaking as a drug. We know the compound is used in alien, which is here. We have the number of vegetable agriculture university also. So to say that, you need so many things. Next one. So what to do is, whenever you make the any Ayurvedic medicine, modern medicine, whatever medicine, you have to do three things: quality, safety, efficacy. Three things are very important. You have to maintain quality, efficacy, and safety. These are basic three pillars of that. Next one. So pharmacological safety you have to do it. You have to do in vitro, in vivo trial. Limited safety is required, stability is required, but I.S.G.B.R.D. these are required for the, if you want to do Ayurvedic medicine, nobody can stop you. You can do it, you need your data, do some scientific validation. Now time is here, we have to do scientific validation of our Ayurvedic product. Next one. So quality means whatever quality you have. Doctors, Asuka also saying go to Babri, Delhi also, even Lucknow also, wherever you are getting the raw material, you cannot identify as a person unless you have the knowledge of that. You have to go for different, check the quality of that. So you have to do authentication of your raw material, quality of that, whether it has divided a microbial load, any metal, these all things are required when you select the procure that raw material. So this is a QC department, any company who has the responsibility to do the quality control of the raw material. These are things required. What are the limits? As per WHO limit, it has to be clear the raw material to go further for the making the medicine. Next one. So installation very difficult means it requires so many things in that, because particular plant difficulty identification of right speed. You go to market, I would say it's very difficult to identify raw material also. Genetic variability is there. In nature, we have priority level also, cross pollination, so we don't know which species, not today, after 10 years, how it will be there. And variation during season also. Even summer, winter, the compound segment varies. Even you can see tulsi in winter, it is, good smell is there, but in rainy season, you will have very small or little species while it is there. So variation during season is there. Even harvested time is differ. Even you can see mango also, any crop also. The right species, right time you will not get the right type of, like opium. The capsules would have yellow color, yellowish color. Then only. If it is overripe, then again it was you will not get a proper amount of opinion on that. There are a number of experiments have been done. And then of lack of information, whatever the activity we are not reported by our pharmacologists, but we as a drug, we collect the material from the market and we supply, we go for the testing and for the PhD degree also. If you are not well prepared, so you are not getting desired activity also. So we have to do number of things. Okay, how's it time? 5 minutes.
Sir, 1 minute.
1 minute, so I can stop here or conclude it. So this is the market where you can see. Madam, I can have 2-3 minutes.
Yes, sir, please go on. I think online, another speaker is
Okay.
already online already.
So next one. So I mean to say, yes, it's very difficult to identify. They look similar. Next one. So you can go fast. So this is an example where they look similar but they are different. Next one. Even the season different. Winter season and summer season they are completely different. Even ecological variation we have found in Acroscalamus. Next one. Even the variation in the oil content 9.26 to 9.5. Nature has, because of soil condition, environment condition, there are variation also. So how to identify? These are the books which are mentioned. Next monograph of I.S.G.B.R.D. These are the books which you can go and you can have next one. So these are the things we require to identify right material. And finally, I will just go to the last slide. Next one, this is how we can identify macroscopically. Next one. Next one, Brahmi, you can identify. Next one.
Yes, you can check the link, sir.
Next one. Next. These are the books where you can have the all QC marker. We have the I.S.G.B.R.D book also. Next one. This is the product we have made in the India for diabetes. This we have made the hot soup from the Ayurvedic. This is how we have made. So in the end, I will just tell you we have the information, we have the instrumentation, we have the knowledge. Only thing you have to work together with the botanist, pharmacologist, and then deliver it product as per the drug material.
Thank you very much, sir. Thank you for the very informative lecture. Actually, I will also be working on grounding, both of the central anesthetica as well as Bacopa, and I will be in touch with you, sir, for the same. Thank you so much.
Thank you.
And our next speaker is already online for the session. Dr. Sunil Kumar Mukherjee, who is an NHS scientist from Advanced Center of Plant Virology, Division of Plant Pathology, IARI, New Delhi. He will be delivering talk on control of DNA viruses using the tools of CRISPR-Cas9.
Continue with YouTube or continue with the Facebook.
YouTube.
So I'll hit the YouTube.
Yes, sir. StreamYard link is there.
Yeah. But it says that you are entering a broadcast studio.
Yes.
And then continue either with YouTube
Yes,
or
YouTube.
continue with Facebook.
YouTube.
Okay. So how do I access my how do I share the
Sir, it will automatically start. It will
Sorry?
automatically start. Just click the link. StreamYard link.
I have done.
You have to click the StreamYard link, which I have sent it to you just now.
No, that's what I have done.
So what it is?
Now I see my face and the mic is working.
Your mic is working.
What should I do? You should say it says mute, stop, and settings. Display name Sunil Mukherjee, enter studio. So I should hit enter studio.
Yes, sir.
Okay.
Yes, sir. We are able to see you.
No, you are able to see
Yes,
me.
we are able to see you. We are
But I need to
seeing.
share my video. I see the share. Should I hit the share?
Yes.
Okay.
Welcome, sir, for the second day of this Bioscience Congress. We are welcoming you from behalf of Hindustan Science and Technology. So I am Dr. Gohit, Dr. Kashmiraji Gohit, Dean Anand College of Pharmacy, and we are conducting this session, scientific sessions. Should we start the lecture, sir?
You are able to hear, sir?
Yeah, I am able to hear, but I'm not able to get the screen where my presentation is stored.
You should be able to see one icon over there where you can share your presentation.
Yes, that I see. I have hit that. I have hit that part share.
So it might take some time.
See, you can hear
Yes,
see
yes,
me.
yeah, yeah,
I can see.
yeah.
So what should I do now?
सुनो अंशुमन को बुलाना.
না তোমার দেখতে হবে অন্য জিনিস দিচ্ছি Road, should I hit share?
Yes, sir.
Okay. I have done, but there is nothing.
Yes, we are able to see your screen, sir.
Where? No, I don't see
Here.
my screen. I don't see my. I can't
You can
find
open
my
your presentation, sir. Yes, sir. We are able to see now. You can open your presentation now.
Yeah, I'm doing that.
Yes, sir.
So we are ready for the next session. Again, Dr. Sunil Kumar Mukherjee, Emeritus Scientist from Advanced Center of Plant Virology, Division of Plant Pathology, would be delivering talk on control of DNA viruses using the tools of CRISPR-Cas9.
Right. Now you are being streamed. Good, sir.
Yeah, but how do I access my presentation?
So now you just
मैम मैम
ये म्यूट कर दीजिए, ये तो म्यूट कर दीजिए जिससे यहाँ की आवाज़ नहीं आएगी। Yes, sir, you are able to hear?
Session, are you ready for the lecture?
Yes, sir.
Dr. Sunil Kumar Mukherjee,
Hello,
Embryo Scientist from Advanced Center of Plant Virology.
can I start?
Please, sir.
Yes, sir, you can start now.
Okay, thank you.
Yes, sir.
The title of my talk, is control of plant DNA viruses using tools of CRISPR-Cas9. And I would like to thank the organizers for bringing me here. And since CRISPR-Cas9 has revolutionized biology, it needs to be discussed. Okay, let's consider a big piece of DNA, and for purposes of genetic engineering, you need to cut the DNA at a desired place. And what are the tools? One tool is restriction enzyme, like E.Q.A.D.1, BAM-H1, etc., etc. But let's say it's a BAM-H1 or whatever, then it's a 6 cutter. So there is one site here. And where is the next site? It would be 4 to the power 6 base pair away. It means on the left or right, if you go about 4,000 base pairs, then you might hit another restriction site. But then if you want to cut in between, what are the options? Okay. So the options are at least 4 options. You can have meganucleases, zinc finger nucleases, talon, and CRISPR-Cas9. Out of these 4, CRISPR-Cas9 is the easiest tool. So now I need to tell you what is CRISPR, what is Cas9, and how do they function. So it all started, surprisingly, with a bacteria and a bacteriophage. The story is just like this. The discoverer of the CRISPR, is Francis Mojica. He is a professor in Spain in the Alicante University. And while he was doing his PhD degree, he's the original discoverer. While he was doing his PhD degree, he sequenced some archaebacteria of his interest and found that in the archaebacteria, there is surprisingly some repeats which are interspaced by spacers. The repeats are palindromic in nature of about 30 nucleotides, and the spacers are also of the same size. The number of repeats and number of spacers later on turned out that it could be different among different bacteria. But then he finished his PhD degree, went for his postdoc, came back, and completely engrossed himself in these sequences, because what he found that in most of the archaebacteria, this kind of repeats and spacers pattern, which was later called CRISPR, is conserved. And in half of the eubacteria, the same thing is present. And since they are separated evolutionary by quite some time, so this is evolutionary conserved. So that interested him, and he followed it up. He wanted to know what are the functions of the spacers and the repeats, etc., etc. So remember, in his time there was no PubMed, so he sequenced his things. He sorted through his things manually, and found that in one of the bacteria, the spacer sequence match exactly with the bacteriophage sequence. Some part of the bacteriophage matched the spacer sequence exactly, and the bacteria was resistant to that particular bacteriophage. So he came up with an interesting speculation that the spacers of this repeat and spacers, which was later called CRISPRs, the spacers of the CRISPRs must be saving bacteria against the bacteriophages. So that was a brilliant speculation, and he sent it out to journals like Nature, Science, but it was rejected on the plea that he has no mechanism. So he published this thing, in some inferior kind of journals, and his speculation remained unheeded to the scientific community. People, came up with many different explanations, like it could be RNAi of the bacteria, and so on and so forth. But all the speculations weeded out in course of time. Only his thing stayed. People found out later on what is the mechanism, and now the field has advanced so much that the original discoverer has taken the backseat, and the people, these 3 people here I show, they are the modern faces of CRISPR-Cas9, and 2 of them, Doudna and Charpentier, bagged Nobel Prize for their work. See, bacteria, if you count the total types of bacteria, and the number will be almost infinite. And they come in different sizes, different shapes. And in a paper in 1998, long back, they calculated that the number of bacteria are 4 to 6 into 10 times 30 is not coming here. Okay, so there are so many prokaryotes, and what is the human population? It's only 8 into 10 to the power 8. So the number of bacteria are huge and huge and huge, and these bacteria are separated by about 3 billion years. Okay, these bacteria are present in soil surface, the sea surface. If you go a little bit below the sea surface, then kind of bacteria you find are mostly archaebacteria. Okay, and these bacteria grow and die, and probably they grow at a rate faster than they die. So the number of bacteria will be much more than the paper showed. That is, it could be that was our 6 into 10 to the power 30, and I believe it will be higher than that now. So as the bacteria are different, the CRISPR-Cas organization is also different. On the upstream of CRISPR-Cas sequences, there are protein sequences, protein ORFs. Okay, and depending on the nature of the ORFs, the CRISPR-Cas sequences are grouped, and they are grouped in 6 different types. And the type today that we will be discussing is Cas9, belongs to type 2. Okay, but whatever the types are, all kind of CRISPR-Cas has to perform these 3 different activities: adaptation, CRISPR sequence biogenesis, and then interference. Maybe I will discuss that part a little bit in detail with the type 2 sequence, the Cas9 sequence that I'm going to talk to you. See, focus your attention on this part. Here you will see the YOLOs, and the YOLOs are the repeats, and then intervening the YOLOs, there are different colors. One is, here it is pink, here it is blue, and so on and so forth. Okay. Now there's one more thing that I need to tell you, that on the upstream of that, there are protein sequences. Okay. And these proteins are required for the CRISPR function. And even in the upstream of these proteins, which are called Cas proteins, there is a site which makes RNA abundantly, and they are called tracer RNAs. And the tracer RNAs has kind of sequences which are half repeats, and actually they complement the repeats which are shown here. So they are anti-repeats. If a bacteriophage attacks this bacteria, then some part of the bacteriophage, shown in blue here, will be cut out by the CRISPR proteins, which are Cas1 and Cas2. Okay, and then after cutting it out, they will put this sequence right at the 5' end of the repeats. So in that way, that sequence is put in, and once that it is put in, the adaptation stage is over. What the bacteria will do now, will transcribe this sequence, the whole CRISPR, and that is called pre-CRISPR sequences, pre-CRISPR mRNA. The Cas sequence will also be transcribed. Accordingly, you will get the Cas proteins, and also this part will transcribe, and you will get the tracer mRNA. Since the tracer mRNA has some complementarity with the pre-CRISPR mRNA, they will form a hybrid. Okay, and the hybrid with lot of mismatches inside. This hybrid then will be taken over by the proteins inside, which are Cas9, RNase3, and few other proteins. They will cut the long RNAs into small pieces. The tracer RNA will be cut, and this is the mature tracer RNA, which is about 75 nucleotides. and then CRISPR RNA will also be cut, and of the size will be 35 to 40 nucleotides. So these are mature CRISPR RNA and mature tracer RNAs, and this part is called biogenetic part of CRISPR RNA. Now, once that is done, this CRISPR RNA, mature CRISPR RNA and the mature tracer RNA, since they have complementarity, they will form a complex like this. Okay, and this complex will be then inviting a protein which is Cas9. See, this tracer RNA interacts with Cas9, and this whole RNA complex, 2 RNA complex, will be further complexed by a protein which is Cas9. And then, if the same bacteria or same bacteriophage or bacteriophage of similar sequence invade this bacteria, then this RNA protein complex will attack the same place, the blue place from where it was cut, and this blue place will be cleaved completely by this Cas9 and 2 RNA complex. Okay, and once the bacteriophage DNA is cut out, then the bacteriophage is dead. So that's how this sequence protects the bacteria against these bacteriophages, and that's the mechanism. And if you look at this RNA complex, it has the cRNA and also the tracer RNA. And the cRNA actually tells the complex, the whole complex, where in the DNA it has to go, because it has the complementarity. And this other part, the tracer RNA part, binds tightly with the Cas9. Okay, and the Cas9 of many different bacteria are of different sizes. But if you take the common one, that is the S. pyrogen, then you will find its molecular weight is very high. It's about 160 kDa, and it consists of many different modules. Okay, and this CRISPR-Cas9 system was later on applied to eukaryotic system, and the most prominent face in this scenario is Jennifer Doudna, who received the Nobel Prize. She not only applied it to eukaryotic system, she modified the 2 RNA complex into 1, and let me come to that part. So she did modification of the Cas9 system. The Cas9 is originally, as I told you, it consists of a protein, Cas9, and then the 2 RNAs, which are CRISPR RNA and the tracer RNA. So these are the 3 components. What Doudna did is that the CRISPR RNA she trimmed and knew what is the minimum size is required of the CRISPR RNA. Okay. Similarly, she trimmed the tracer RNA. The minimum size of CRISPR RNA was 20, and the minimum size of tracer RNA is about 65 or so. And then she joined these 2 separate RNA by a loop of 3 to 4 nucleotides. And in that way, she made a combined RNA, which is about 90 nucleotides, and termed it as gRNA. Okay, so gRNA consists of 2 parts: a part of CRISPR RNA, which takes the Cas9 protein to the DNA at the specific site, target DNA at the specific site, and the tracer RNA component binds to Cas9 very, very tightly, and that's how it works. And this whole complex, you can use it to cut DNA at any desired place you want. Only thing is, you have to keep this the DNA targeting sequence to the right target. Okay, so this is the system which allows you to cut DNA at any desired place you want. Okay, so let me illustrate this again, whatever I said, and how this CRISPR-Cas9 system can be exploited in the eukaryotic cell. So in the eukaryotic cell, you introduce this Cas9 and then the gRNA. Okay, they will form a complex, and while they make a complex, the Cas9 protein, which is normally inactive as a nucleus, as a DNase, undergoes many allosteric changes, and then this part, with the help of the spacer RNA, finds where in the target DNA it has to attach. So it goes and attaches, and while attaching, there is a site called PAM site where the whole complex, the Cas9 complex, gets attached with very, very tight affinity. And once it attaches, so this is the spacer part which has hybridized with the target DNA, and since the DNA is duplex, the other part loops out. So this is called a RNA loop or R loop. So it has a hybridized RNA DNA component and a looped out RNA. So this is an RNA loop. This RNA loop is then cut by Cas9 3 nucleotides upstream of the PAM sequence. Okay, so it is cut. It is just like this. So up to this, and it follows whatever happens in the prokaryotic system. But in eukaryotic system, something additional is happening. So these two broken sites are then protected by proteins which are called repair proteins. And a protein called Ku is an important component of the repair machinery. And this repair machinery wants to join these two broken ends, and they do. And while joining, some mistakes are done. By mistake, I mean there could be some deletion here, some insertion here, some small mutations here, and all this. So this is called non-homologous end joining. So at the broken end, either sequence is deleted or added or mutated. Okay, so by non-homologous end joining, this broken DNA gets joined. In other words, this whole DNA gets mutated because it has lots of mutations inside. And the other way to repair is that you can introduce a sequence of your interest at the broken end by homologous recombination also. So these are the two ways. This is called HDR, homologous dependent repair, and this is non-homologous end joining. So using these two techniques, the whole broken DNA is repaired. So that's how the Cas9 interaction with gRNA happens. The thing in the ash color, whatever you see, is a Cas9 protein. And in the yellow, whatever you see, the target DNA with which the gRNA can attach, the spacer part of the gRNA can attach. Okay, and this is the other part. See, if you look at the target site, so there are about 20 nucleotides, which are the target sites, and this is the tracer part. Okay, this tracer part is shown here in the red, and it attaches the Cas9 very, very tightly. So in other words, in this crystal structure, you will find the whole protein can be divided in two parts. The top part, which is called a recognition site, where the tracer RNA recognizes this component of the protein and attaches very tightly. And then the other part, which is shown here, this is called nucleus part. There are two lobes, you see. There are two structures over here, this structure and this structure. And this structure is called HNH, and the other structure is called RABC, and both of them has DNase function. So as you see, the Cas9 protein has grabbed this part, so this part will be cut into two parts later. So that's how the DNA protein interaction happens with the Cas9. Okay. So the question is, what is the consequence of this kind of repair? Okay, the consequence is, the first consequence, if it happens by non-homologous end joining, then the DNA or the gene in question will be inactivated because there will be mutation. Okay, so what is the purpose of this inactivation? Can you use it? The answer is yes, because you will find that there are many genes which act as a regulatory protein. For example, the rice grain. The rice grain size is of a specific size. Okay, it is not very big or not very low or very small. It has a specific size. Now, what makes this size? Which protein makes this size? And that is a rice protein called SPL4. Now, since it makes this kind of size, if you inactivate it, then you can expect the rice grain to be larger, and you can that way increase the rice yield also. Actually, that you can do, that has been done in purpose. Okay, and if you cut the fruits like apple or mushroom, you will find that after cutting, the cut surface become brown. It happens because there is a protein called polyphenol oxidase, which in contact with oxygen, makes lot of phenol, and that phenolics gives the brown color. So now if you inactivate that PPO protein of mushroom, then there won't be any browning at the cut surface. And that, this browning resistant mushroom are actually sold in the market. Okay, let me illustrate the next one. Homologous recombination. We're using homologous recombination. Now you can use a DNA part of your interest. Now let's think of some genetic disorder. So there, the normal genes are not there, they are mutated, and if you want to correct it, then you can cut that DNA using CRISPR-Cas9 and introduce a new functional piece of DNA. Okay, in that way you can correct the disorder. And people are born with birth defects. There are different kinds of mortality, and these are all due to having bad genes. But you can convert the bad genes into a good gene using CRISPR-Cas9. So shut off, or you can make the gene hyperactive using this, and then you can genome edit using CRISPR-Cas9, and there are many other applications. And as far as today's talk is concerned, I will confine myself to the genome editing part. Okay. And genome editing, genome of almost all, like human being, animals, plant, all genomes have been edited, and most of the editing has done in China, and that they have done in the agricultural field, in the plant field. Now we will come to that, but before coming to the application of CRISPR-Cas9 in plants, I like to tell you about a human application. Now CRISPR-Cas9 has come to the clinics. It can solve the human diseases. There is a disease which you might heard, which is called sickle cell disease. in India, in Africa, and in many places, in Europe, almost everywhere, uh, newborns take birth with their blood cells which are defective. Okay, in the blood cell there is hemoglobin, and hemoglobin is a tetramer of 2 alpha chains and 2 beta chains. And in this kind of patient which have sickle cell disease, in the beta globin gene there is point mutation, from A to T, and then there is a change in amino acid in the beta globin gene from leucine to valine. And when that happens, the normal beta globin gene or normal hemoglobin becomes sickle cell shaped. This is the normal one and the normal RBC, but this is the sickle RBC and this is the sickle hemoglobin. Okay, and what is the consequence? When the hemoglobin is sickled, this all hemoglobins clustered together. And when they cluster, blood cannot flow across, and whatever the organs downstream, they are deprived of blood and oxygen. So the organs cannot function. And as a result, the patient gets a lot of pain in that organ. And these are called pain crises. And most of the patients die prematurely. So this is a very, very severe disease. And how to handle that, because this disease is around for a long time. There are many ways you can handle, but the best way to handle is to give good blood to the patient, and that's called transfusion. But the transfused blood cannot last long, so you have to keep doing it in order to keep the patient surviving and well. So you have to keep doing it. So question is there any permanent solution? The CRISPR-Cas gives permanent solution. Okay. How do you do it? That's a big story, and I will try to be as brief as possible. Before birth, before coming out from mother's womb, all the newborn have a hemoglobin of type which is called fetal hemoglobin. Okay, and the fetal hemoglobin actually then later on is repressed by a protein as the kids take birth. Okay, and a new kind of globin gene, beta globin gene, comes up. So when they are born, kids are born, they have mostly fetal hemoglobin in very less amount, and then beta globin gene in a very high amount. But in the patient, the beta globin gene is defective. Okay, so question is how to correct it. So the repressor in which represses the fetal hemoglobin gene is cut. And if you cut it, then the fetal hemoglobin will be made in large amount, and that will continue within the patient. Okay, and this kind of medicine or whatever you call is called CTX001. And the people who have taken this thing, CTX medicine, their blood is enriched with fetal hemoglobin. So they don't have the sickle cell hemoglobin very much. And as a result, they survive. The first successful experiment was done with a patient named Victoria Gray in United States. And that was done in 2019. And from that time onwards till now, she has not gotten any pain crisis, or she did not require any other blood transfusion. So one-time blood transfusion did hell of a good job for her, I'm sorry. And then after her, there are 22 other patients which got the same medicine, and they're also all okay. So to have that medicine, hundreds of patients have now registered for this treatment. So this is a great discovery, I would say. because it has come to the clinic already. So this is about the human condition. This is about the human condition. How about plant? Now, plant has also a marketable product. Tomato makes a short, I would say, short fragment which is called GABA, which acts as a neurotransmitter, and it also controls our blood pressure. Now, in the tomato, GABA is made, but GABA is also cut by regulatory proteins. So that regulatory protein, if you do CRISPR-Cas on that, then that protein will be inactive and GABA will be made in very high amount. Okay, and this GABA overproducing tomato is being sold in Japan market, and also they have been allowed to come into European market. Okay, so regulatory permission has come in. So CRISPR-Cas has also a marketable product, and not only that, there are many marketable products which might come in a span of another 5 to 10 years. They are in the phase 2, phase 3 trial, like cherry tomato. Using CRISPR-Cas, you can make it large, you can make it high vitamin content, and they are also resistant to the bacterial spot disease. This white button mushroom I have told you already, they are browning resistant. And similarly in rice, orange, cacao, wheat, this has been CRISPR-Cas has been used for many different uses. Like in wheat, gluten is a great problem. Like in wheat, gluten is a great problem, and there are 30 to 45 genes which have been manipulated so that the wheat becomes low gluten. But professionally, we are plant virologists. We like to make food which are free of plant viruses. Plant viruses are of two different kinds. One is DNA virus and the other are RNA viruses. I am not going to talk about RNA viruses at all, but we'll talk about DNA viruses. We'll like to give you the strategy how to make the plants free of DNA viruses. And what are the different DNA viruses? There are several different types of Yemeni viruses. If you look at viruses as a whole, then there are two large groups. One is Yemeni virus group, and the other is virus group. Is RNA virus, and the Yemeni viruses can be divided into 13 different genera, and each genera has their own typical viruses. Okay, typical species. So there are many different types of Yemeni viruses. There are cauliflower mosaic viruses. Any crucifer is affected by cauliflower mosaic viruses. Banana is affected by streak mosaic viruses. I'll come to that part in a little bit later. And you can get rid of these viruses using two different means. You can allow plant cells to express Cas9 protein and the gRNA which target the viral DNA. In that way, viral DNA would be mutated, would be inactive. So, plant cells will be gotten rid of the viral DNA. That's one approach. The other approach is within the plant there are genes called sensitive genes, S genes, which normally help the viruses to grow. And if you detect the sensitive genes, then if you inactivate or mutate the sensitive genes, then the viruses will have hard time to live in the plant cells. In that way also, we can make plant cells resistant to the viruses. Okay, just very small introduction about the Yemeni viruses. If you look in the electron microscope, they will look like geminate particles. The two particles joined together, like twin. There are twin brothers, which gets conjugated during their birth. So these are the geminate particles, And these Yemeni particles cannot infect plant by on their own. They need a vector, and the vector is a very small white fly. Okay, and when they infect plants, you get a range. Then, since there are different types of plant, you also get a range of disease symptoms. Here you see the common bean, which is used as a staple food in South America. You have golden mosaic disease. The tomato will have leaf curl, and so on and so forth. Here you have potato mosaic virus. The mosaic symptoms are here. So they cause actually huge loss in the agricultural field. The amount that I show here is actually an underestimate. This is few years old. In India, it should be more than now, 2,000 million US dollar. Pakistan is actually a great sufferer because their cotton is affected by cotton leaf curl virus, which is a DNA virus, and the amount of loss is humongous. In the USA also, the loss is underestimate here. It will be much more than this. And then cauliflower mosaic virus. The cauliflower that comes in our Indian market, at least 10% of them are infected by cauliflower mosaic viruses. They don't do much harm to human being, but these are infected. But this cauliflower mosaic virus, hits Britain and France, especially cauliflower of it, to a huge extent. And these are the electron micrographs. That's how they look like. So they look like a ball, and there is double-stranded DNA inside, and these are the core proteins, and they are transmitted by aphid vectors. And then banana streak virus. So the leaf here, the banana leaf here looks healthy, but these viruses stay integrated within the chromosome of banana. So when there is a stress like drought stress, heat stress, flood stress, etc., those viruses which are integrated, they come out and gives the streak-like appearance in the leaf. And from this leaf, you can isolate the banana streak viruses. So the amount of loss they make, since it is restricted to banana, is not much, I would say. So out of these three different types of viruses, Yemeni viruses are the one which affects the agricultural field to a great extent. So most of the papers which showed that removal of Yemeni viruses are possible from the plant are with Yemeni viruses of different kinds. But I would like to make a specific observation, covering all these experiments, which wants to get rid of Yemeni viruses. See, first challenge is that the Yemeni viral genomes, when they enter in the plant, they enter as a single copy, but they multiply in thousands. So the target size is huge. So you need to express Cas9 and gRNA in a huge amount by a strong promoter. So that's the first challenge. And even if you make them in high amount, even if they cut the viral DNA, the viral DNA can rejoin, and in that way they can make new viruses. They would be of low copy, but they can make new viruses. So these are called escape mutants. So the elimination experiment is bogged down with escape mutation also. And of course, you want to target the Cas9 at one place, but Cas9 can also see similar places in the chromosome, and thus they can have off-target effects. So these are the challenges that this kind of experiments have. So the essential principle for inactivation of the viral DNA is just like this. This is the Yemeni virus. It comes in the plant nucleus as a single copy. It multiplies, it immediately is converted to double-stranded DNA. Okay, and host factors are involved for that. And then it multiplies, it replicates and gives you 1,000 copies. And after that, they make lots of viral protein, and those viral proteins make the plant diseased completely. Now, if you can express Cas9 and gRNA in the plant, then the gRNA will target the double-stranded DNA, which is made here, double-stranded DNA at a specific place, guided by the gRNA. And then if it targets, then it will be cut, and once it is cut, it cannot replicate further. Okay, and even if they are rejoined, the replication is almost impossible, but sometimes possible. But anyway, this is how you get mostly the resistant plants. You need to tell me if I have to stop somewhere. I will give you the case study, one case study. That is the tomato yellow leaf curl virus. And that is, this virus is very much predominant in Europe, and especially in Israel. In Israel, you cannot grow any tomato without growing the tomato yellow leaf curl virus. Okay, and this virus can be inactivated using the CRISPR-Cas9 sequence. How do you do that? This is the genome of TOIL-CV, the viral DNA, and in the red area, you make gRNA so that these areas are targeted. And once these areas are targeted, then the viral DNA is eliminated. I'm not showing the details here, as I see that the time is running out. But if you sequence the joint viruses, you will find. Hello.
There is still 5 minutes now. It's 5 minutes for the lecture. 5 minutes remaining for the lecture.
All right. If you look at the sequence, wild type sequence, you will find that this is the area targeted, and after targeting, there is deletions and there is some mutation also. In that way, this CRISPR-Cas cuts the DNA and make the plant resistant to the viruses. Okay. And there are sites where targeting is more effective than others. Like the target, where the replication is initiated. If you target that site, then the targeting is most effective, and that's how wherever the Yemeni viruses have been attempted to be eliminated, that site has been targeted mostly. But in spite of that, there are off-target effects and the problem of escape mutations. So how to get rid of escape mutations? And we did some work ourselves. So we thought that, instead of cutting at one place, if you cut that at two places in the viral DNA, so that when they get rejoined, it would be very hard for the virus, to be an effective, productive virus. So there are many others. This is Dr. Anirban Roy and myself. This is Dr. Anirban. This is Bikash Mandal. So as an experimental system, we took the chili leaf curl virus, and the experimental plant was Nicotiana benthamiana. Okay, so we identified the sequences first, which to be targeted, and in the IR region, you see there are many targets, but if you target the black ones, then there is possibility of having more off-target effects. So by bioinformatic means, we selected the right kind of targets. In the IR, there are two targets. In the rep protein area, rep protein is the protein without which virus cannot replicate. So these are the other two targets. And the coat protein also, there are two targets. So in total, we got 6 spacer targets, and then wanted to see whether they're fruitful, they're effective. So we took a span of DNA, which is about 1.5 kb, which has 3 different targets, the IR targets, coat protein target, and the rep protein target. And then we gave the gRNA and the Cas9 and wanted to see whether, the DNA is cut right in vitro. As you see, they cut. They not only cut, if you give them in combinations, one gRNA plus two other gRNA, then they also cut right. So all the gRNAs that we made are good in cutting viral DNA individually or in duplex conditions. So the question is, do they work in plant? The answer is yes. I don't have time, to describe the whole thing in detail, but you see, if you give these two gRNAs in combination, that is gRNA 5 plus 4 and gRNA 5 plus 2 and gRNA 1 plus 2. 1 plus 2 is this, and 5 plus 2 is this, 5 plus 4 is this. Then, the virus titer, the virus DNA is almost minimal. And why these two combinations are effective? Because they make more gRNA than others. And we looked at the symptom formation using several different gRNAs that we made. And when we use 1 plus 2, there is a bit of leaf curl, of very short, small leaf curl, and then there is stunting. But this gRNA, 5 plus 2 combination, there is no leaf curling, there is no stunting. So here, the virus was almost eliminated as far as symptoms are concerned. Okay, so these are the symptoms at the local site, and the similar story with the symptoms which are at the upper side, where there was no treatment given. Okay. Then we wanted to check whether there's an escape mutation formation. Okay, and there is a slide which I would take time to explain, but our assay says there's an exonuclease T7-1 assay. It says that there is no escape mutation in this kind of experiment. So the multiplex conclusion says that, if you have the right kind of combination, you can eliminate the virus altogether. So this is one story, and this is a table which shows that many gRNAs and the banana stick virus and cauliflower mosaic stick virus has also been eliminated from the respective host. So there is another story also. How about the S genes, which allow virus to grow? So if we eliminate the S genes, then the virus will have hard time. In RNA viruses, the S genes are known very well, but in DNA viruses, they are not known. But recently, there is a gene which is called pelota, which keeps mRNA intact. Now, this pelota has been shown to be helping the viruses. Okay, so if you inactivate pelota of tomato or the pepper, or in fact this is a very conserved gene of any gene, then you will find that the DNA viruses from those plants will be eliminated. Okay, this is the tomato story. And this is the CRISPR-Cas applied to the tomato and the first section if you do, and then you'll see the virus titer is almost zero. So there are two approaches. Both approaches needs to be applied. So my final conclusion is the viral DNA can be directly or indirectly eliminated by CRISPR-Cas9 system, but there are bottlenecks like off-target effects and escape mutations. And once these bottlenecks are taken care of, you will get virus-free food in the market. And there I stop.
Thank you very much, sir, for the very, very technically useful lecture. Pharmacogenomics is coming up very, very useful nowadays, not only in terms of genetically modified food, but also the individualized medications for human being, which will be very, very effective in the coming years. So we have biotech audience also, and
So if you have any question, I would like to answer.
we can take the email ID also.
Yes,
And
sir.
Yes,
in
sure.
that case, you can get in touch with the speaker for your. We have the next
The
speaker
lecture,
online.
the lecture would be, sir, online on YouTube, and I think the students can listen it again and again. You have explained the entire mechanism in so detailed way that one has to listen one or two times. I have also a few questions. I would there are many questions, but I will just ask only few. So one is the thing is that you explained it that we can do the genome editing, but in the case of wheat, if there are three genomes like A, B, and D, then exactly what is the way if we want to do the specific
See,
editing like
if the gene we are targeting, has many isogynes, then you need to look at the conserved site.
yeah.
And if you hit the conserved site, then all different isogynes would be made dead.
And if we want to do the deletion exclusively in one genome, then we have to do the other way around.
Yeah, right. You have to look at the non-conserved sites.
And sir, second question is the structure of the enzyme. The Cas9.
Yes.
Is it conserved or is it?
See, the modules that I have shown you, like HNH, RABC, okay. So the RNase component of those are conserved, but in different bacteria, the intervening sequences are different.
Okay. So is there any other questions from any other students? We are running out of the time, and I would request, yes. Okay, so there is one more question.
So one more
Yes,
question.
please.
Yes, sir. Student, how a student thinks, I'm asking. You have told about that.
Could you be a bit louder? I can't hear you.
Okay, sir. You have told about that viruses, Whisker Cas virus can kill the sickle cell.
So what is the question?
No, I'm just asking you. Hello.
Hello?
Can you hear me, sir? Can you hear me?
No, I can't hear you. You have to be a bit louder.
Okay, can you hear me now, sir?
Yeah, it is much better now.
Okay, thank you. It's a question from the student side I'm asking. That
Yes.
You have told that this virus, this CRISPR-Cas gene or enzyme is treating the sickle cell anemia. Can this also be used for thalassemia patients in the near future?
Can it be used for what?
Thalassemia patients, because it's also a hemoglobin disease.
So what are the patient's name?
Thalassemia.
Yeah, beta thalassemia.
Yeah.
Yes, absolutely. You use the same technique, exactly the same medicine to treat beta thalassemia.
Okay.
Yeah.
And one more question, ma'am. Can I ask? Yes. Okay. You have told about that escape mutations are going on in virus.
So I can't hear you.
You have told about escape mutations.
Yes, escape mutation.
Escape mutations going on by cutting a single site. Is it so?
Escape mutations? Then what is the
Escape
question?
mutation occurs in the virus if we cut the virus genome at a single site. Is it so?
Yes, if you cut the virus at single site, the probability of escape mutation is much higher.
Okay, thank you.
But if you cut at different sites, then it is low.
Just one more thing, we are planning to do one gene editing workshop in the month of November and December, and in that also we would be also demonstrating the students the technique over here at our college, and we would like to have you during that time too.
Yeah, it will be my pleasure. But you have to,
we are very thankful that you are here, sir. In spite of the thing you have to go, and so we are really thankful.
Thank
No,
you,
I
sir,
am
for the
also
wonderful lecture.
doing my patient
This is an
presentation.
immensely helpful lecture, and we'll definitely get back in touch and email with you.
Sorry?
We will get in touch with, on email, sir, because this is an
Yes, yes,
immensely
yes,
related pharmacogenomics. It is a very, very helpful topic, as 9 and all. So
yeah,
the lecture,
sure.
as already sir said, is available on YouTube. So student as well as any delegates could see it anytime they want it. And
Yeah,
they will definitely get back in touch any queries on your email IDs. Thank you so much.
most welcome.
Thank you. We are ready for the next session,
He is joining.
which is our speaker is online already. Professor I.S.G.B.R.D, who is the president of German Association of
Yes,
Homotherapy
sir, I'm waiting
Germany,
you to join.
will join us for this session, and he is already online. We
Hello,
welcome
yes,
you,
sir, I'm
sir.
waiting you to join.
We welcome for the next lecture. Audience can interact very well. I mean, if you are very, very actively interested, I mean,
Okay, good, good, good.
good afternoon. Good afternoon, sir.
You are audible, you are audible.
30 minutes.
Dr. Burns, we are welcoming here in this session. We just completed one session and we are ready for you right now. I'm Dr. Kashmira Gohil. Session. We just completed one session. We are ready for you right now. I'm Dr. Kashmira Gohil. We are ready for this session, sir. Please start your lecture. We are ready for this session, sir. Please start your lecture. We are ready for this session, sir. Please start your lecture.
So online?
Yes, sir, you are online.
Okay, thank you.
Please close.
Online? Thank you.
Ma'am,
Online? 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 Something wrong with the out. Something wrong with the something wrong with yeah. Yeah.
It's running somewhere in the computer or
Yeah.
somewhere else.
Yeah.
It's running in the media.
Yeah. Okay. 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 Hello. Good afternoon. Good afternoon.
Good afternoon, sir.
Is it okay now?
Is it okay now?
There's still some
There's
echo.
still some echo. Sir, it's good now. It's good. It's good now, and we are continuing with the session. Please continue, sir. Please continue.
Okay.
Okay.
So good afternoon,
So good
everybody.
afternoon, everybody.
My name is Ulrich
My name
Berg,
is Ulrich Berg,
and the
and
subject
the
of my
subject of
presentation
my
today is
presentation today is
Agnihotra and Cobra
Agnihotra and
farming,
Cobra farming,
tools for a
tools
sustainable
for
development
sustainable
on planet
development
Earth.
on planet Earth.
Now,
Now,
there is some
there is
echo.
some echo.
It's very difficult. So, the challenges of our present situation are the following. See, science is the search for truth, but in order to help solve the problems of our present time. And the most prominent problems which humanity faces today are environmental problems, pollution, and climate change. Biosciences can help a lot to mitigate these problems and help make changes for sustainable development of our planet. Such a sustainable development was discussed by United Nations in 2015, and they adopted 17 sustainable development goals, also known as the global goals, as a universal call to action. Aim was to end poverty, protect the planet, and ensure that by 2030 all people enjoy peace and prosperity. These 17 sustainable development goals are integrated. They recognize that action in one area will affect outcomes in others, and that development must balance social, economic, and environmental sustainability. In this presentation, I cannot go and cover all 17 Sustainable Development Goals. So I will concentrate on the first 3 goals: no poverty, zero hunger, good health and well-being. And as these goals are interconnected, working towards the 3 goals will also have an effect on all the other Sustainable Development Goals. In order to achieve these 3 goals, no poverty, zero hunger, good health and well-being, first let us look a little bit more at the problem which pollution creates. This map shows the percentage of pollution-related deaths, and you see in some areas it is not so high, 0 to 5%, like in US and some countries in Europe, also in Russia. But in other places, like in India, we have 20 to 27% of all deaths are related to pollution. So that is really a big number, and that shows how much humanity is affected by this pollution. But it's not only the pollution, animals are suffering as well. And just one example, a study 2 years ago in Germany showed that there was a reduction in insects of 75% within the last 25 years. 75% less insects in the last 25 years. The scientists who did the study commenting on the results that that's a big, big catastrophe. So humans are affected, animals are affected, but also plants are affected. Just one example, the air pollution damages leaves. So that makes plants more vulnerable to pests and diseases. Photosynthesis is reduced, growth is reduced, and the yield goes down by 5 to 20% according to recent studies. That means all our life on this planet is affected by this pollution of our environment, soil, atmosphere, and water. And that is a tremendous challenge. And what are possible solutions? So one of the solutions which I want to present here is Agnihotra Entomotherapy, which comes from Vedic knowledge. What is homotherapy? It is the science of feeling the atmosphere through pyramid fires and eliminate pollution and contamination. The smallest and basic Homa healing fire is tuned to the specific biorhythm of sunrise, sunset. It comes from the ancient most Vedic sciences of bioenergy, medicine, agriculture, and climate engineering. So what you need for Agnihotra, one thing is a small copper pyramid of fixed size and shape. You see it here. It has 3 steps. You need some dried cow dung, some pure cow's ghee, and some unbroken, unpolished rice grains. Then you need some Sanskrit mantras, exact vibration, and timetable of sunrise and sunset exact to the second. Why these mantras? Why exact timing? In the ancient tradition, sunrise is described as follows. At sunrise, the many fires, electricities, ethers, and more subtle energies emanating from the sun extend all the way to the earth and produce a flood effect at those coordinates where the sun is said to rise. Interesting, they say it is said to rise because they knew it is not actually rising and setting. And this flood of energy carries a subtle music in it, and the quintessential sound of that music is the mantras which we utter at that time. Therefore, it is clear these mantras are important and they have to be exact. If you translate, you miss the effect. And the time is exact, important, because this flood of energies lasts only for a very short time band. Interesting is some American astrophysicist made this sketch, and he showed that at sunrise, he says million ampere currents head into the Earth at sunrise and out again at sunset. So he confirmed this ancient Vedic knowledge. The mantras, 2 simple mantras in the morning, 2 simple mantras in the evening. In the morning, it's Suryaya Svaha, Suryaya Idam Namama, Prajapataye Svaha, Prajapataye Idam Namama. And in the evening, Agnaye Svaha, Agnaye Idam Namama, Prajapataye Svaha, Prajapataye Idam Namama. And as I said, these are exact vibrations. So we have to do the mantras in exact that way. You can translate, but you cannot utter the translation. That will not be Agnihotra. So what happens? We see you perform Agnihotra, and from the pyramid, from the fire, a channel is created through all the atmosphere. Prana energy, life energy, which lies above the atmosphere, comes down through this channel, and an aura energy field is created all around all plants nearby. So these plants get nutrition, they get disease resistant, they get stronger. When the flame dies, all the energy comes back and is collected in the Agnihotra ash. Therefore, this ash can be used as medicine for plants, nutrition for plants, for many, many purposes. So Agnihotra then purifies our environment, atmosphere, water resources, and soil, and thus brings nature back to harmony. And I said this method, Agnihotra Homotherapy, this method comes from Vedic knowledge. But we do not just take Vedic knowledge for granted. Oh, that is written in the Vedas, therefore it must be true. No, we take that as hypothesis and check whether we can confirm with methods of modern science these ancient Vedic statements. And so let us look first at the atmosphere. Interesting is that according to ancient science, but also according to biodynamic farming, for example, it is said that more than 75% of the nutrition which plants get, they comes from the atmosphere. So means if the atmosphere is polluted, plants do not get the nutrition they need. But if the atmosphere is purified and enriched by all these healing fires like Agnihotra, then a type of protective coating comes on plants, and diseases, fungi, pests, and so on do not thrive. And the plant's capacity to breathe increases, and photosynthesis is increased, so the plants just grow better and are healthier. The purification of the atmosphere, there are different types of pollution of the atmosphere. One is biological, by microorganisms. One is chemical, like SOx, NOx, and one is physical, that is particulate matter. Let's first look at the biological pollution, microbes. Here you see petri dishes with cultures of bacteria. At the left side, the petri dishes were opened, say for 10 minutes before Agnihotra, and then used for cultivation. And at the right side, the same thing was done, but after Agnihotra, and you see the big difference. So there's a big reduction in bacterial growth through Agnihotra. And this experiment was done number of times, published number of times, so that's well established. Now, chemical and physical pollution. Chemical pollution is mainly SOx and NOx, and physical pollution, the particulate matter. We did this experiment in different institutes. One was in North Maharashtra University, and here you see the results. We performed Agnihotra, and right after Agnihotra, the values went up a little bit, but then they came down by 30 to 40%. So This experiment was done in the evening, and we measured then in the morning again, and you see the big reduction. So the professor involved in this study said, oh, maybe it is just because in the night, these pollutants just settle down to the ground naturally. So we did the same experiment from morning to evening, and we got the same results, reduction of, say, 30 to 40% of all these parameters. So we can say biological, chemical, and physical pollution are all reduced by performing Agnihotra. One interesting study was done in Germany. It is about the negative ions in air. We know that lack of negative ions, it's an indicator for polluted air, for air without any energy. And negative ions, higher counts of negative ions, are an indicator for fresh air. So normally, smoke reduces the amount of negative ions. We did this experiment more than 30 years ago in Germany, and the engineer who had developed the equipment to measure the count of negative ions, when I said what I want to do with Agnihotra, he said, oh, will there be smoke? I said, yeah, there will be some smoke. He said, oh, then we don't even have to do the measurement because any smoke reduces number of negative ions. And I told him, and in order to prove his statement, he blew his cigarette, the smoke of the cigarette, in the direction of the equipment, and yes, negative ions came down. So I said, still, let's try and see. So we performed Agnihotra, and immediately the count of negative ions went up. So that count of negative ions can be a possible explanation for the effect of Agnihotra on other types of air pollution, like biological, chemical, physical. Now, this experiment, which I just mentioned, that was done more than 30 years ago, now it is replicated, and the long-term study is in preparation as part of a PhD study in Germany. And here you see the results, some results, and at sunrise and at sunset, there's a big peak because we are performing Agnihotra there. What are the effects of Agnihotra and Agnihotra ash on water resources? One experiment was done in Pune. It was a simple experiment. They took water from a polluted river in Pune. Okay, every river in Pune is pretty much polluted, the Mula Muta River. And they poured this water through a column in a glass cylinder of Agnihotra ash. So a glass cylinder was filled with Agnihotra ash, 20 centimeter high cylinder. And then the water was poured through this cylinder filled with Agnihotra ash, and it was the quality of water was measured before and after. And you see this treatment with Agnihotra ash reduced conductivity by 48%, total solid content by 90%, and so on. And the most interesting, the bacterial count went down by 93 to 98%, depending on the method you measured. So from this very polluted water, you got water which then was in accordance with the standards for drinking water set up by WHO. This is the effect of Agnihotra on a bore well. That was in Australia, one farm. They drilled the bore well. They were lucky enough to get water, but the water was highly alkaline, sodic, and high salinity. pH was 9.5, salinity 1150. So this water just could not be used. You can't give it to the plants, they will just die. You can't, of course, you can't drink it. So what they did is they performed Agnihotra next to the bore well, and once in a week, they added Agnihotera ash to the bore well. And after half a year, the pH came down from 9.5 to 7.2, salinity from 1150 to 720 parts per million. So that was now drinking water quality. So practically their farm was saved. What are the effects on the soil? One thing we have to be clear. Soil is very important. And there's a Sanskrit text written more than 3,000 years ago, nearly 4,000 years ago. And that says, upon this handful of soil, our survival depends. Husband it, and it will grow our food, our fuel, and our shelter, and surround us with beauty. Abuse it, and the soil will collapse and die, taking humanity with it. So our existence, the existence of humanity really depends on the health of soil. Now, if you look at the numbers which FAO, Food and Agriculture Organization of the United Nations, publishes, they say already now 1/3 of the world's soil has been degraded. And they continue, unless new approaches are adopted, the global amount of arable and productive land per person in 2050 will be only a quarter of the level in 1960. And that will definitely not be enough to feed all us people. So the soil needs to be rejuvenated first by homotherapy. And this rejuvenation starts from the level of viruses, bacteria, fungi, algae, and so on. In this way, a healthy microflora and microfauna is created. So we have some experiments which confirm the effect of Agnihotera in this direction. First thing is, you know that phosphate is one of the major nutrients which plants need. So this is one of the NPK formula. And one experiment done in US nearly 40 years ago showed that the percentage of water-soluble phosphate in soil increases if you add Agnihotera ash. Normally, phosphorus is in soil. There's enough phosphorus in soil, but mostly in a non-water-soluble form, and thus plants can take it in. So if you add Agnihotera ash, this non-soluble phosphorus will be turned into soluble phosphorus, and the percentage of water-soluble phosphate increased approximately by more than 4 times. This is what the result showed. Then the question was, the same result, similar result was we got in Germany a couple of years ago in some agricultural research institute. But the question was, okay, how come? How does Agnihotera ash increase the water solubility of phosphorus? So one hypothesis was that it is due to phosphorus solubilizing bacteria. And this hypothesis now has been confirmed by a study done in some college in Ratnagiri, and they showed that nitrogen-fixing bacteria, also very important for the soil health, they increased 100 times by adding Agnihotera ash, but phosphorus solubilizing bacteria, they increased even 1,000 times. And at the same time, harmful fungal flora was controlled. Similar experiments about bacteria and fungi were done at the Central Institute of Subtropical Horticulture in Lucknow. And what they could show is you add Agnihotera ash and harmful bacteria, no growth. But at the same time, beneficial microbes, there is increased growth if you add Agnihotera ash. Now, one other very interesting story. We know that we have in India and other places in the world also areas where the soil is very sodic, high pH. And this was one piece of land in a KVK in Unnao. Unnao is between Lucknow and Kanpur. And the pH was 9.86. So what they did is they divided the area into 3 plots. And one plot was with agrochemicals, one plot was organic with vermicompost, and the third plot was organic with vermicompost plus Agnihota ash. And see what happened after one season. With agrochemicals, nothing happened. With vermicompost, the pH came down from 9.86 to 9.06, which is already quite a good result, which shows we definitely should go for organic farming. But with vermicompost and Agnihota ash, the pH came down from 9.86 to 7.67. So that is nearly neutral just in one season. And then people say, oh, you know, with sodic land, Agnihota ash brings the pH down. But not only that, there was one farm in Poland. Actually, I'm now just sitting at this place, Prugorania in southern Poland. And when they started, the more than 25 years ago, the pH of the soil was 4.4, very acidic. And the government agricultural engineer who came and visited and advised these people, he said, practically forget about farming or gardening, nothing will grow. Unless you add tons of lime, and the people didn't have the money for that. Still, they tried. They added Agnihota ash to the soil, and you see they grow everything, including tomatoes. Nobody in the area here grows tomatoes. So, and later on, they tested the soil again. Again, it was 7.2. So Agnihota works in both ways. It just brings nature back to harmony. If pH is too high, it brings it down. If it's too low, it brings it up. And look at the situation in the world. We have more than 800 million of hectares of sodic soil and approximately 180 million hectares of acidic soil worldwide. So if these lands could be reclaimed and made productive again, that would be a big step forward to help feed the growing number of population. And this is why we suggest, why not start such a small project on either acidic or sodic soil or both? That's a small investment, but the possible gain is huge if you think of all the hundreds of millions of hectares which could be reclaimed that way. At the beginning, I mentioned that this environmental pollution affects plants, animals, and humans. And Agnihota, as I've shown, purifies the environment, atmosphere, water, and soil. And now, what are the beneficial effects on plants, on animals, on humans? Let's start with plants. Plants, some very simple experiments. There's something, you know, even on school level, you can do it. You take in a petri dish with some tissue, put some seeds, and see the germination. Of course, you add water regularly. Left side, it was tap water. Right side, it was tap water with Agnihota ash. But people could object, okay, any ash has some nutrients, so it doesn't show that it's really the effect of Agnihota. So what we do is we prepare something called control ash. We burn the same substances, cow dung, ghee, a little bit of rice, but not in this copper pyramid, but in some other copper vessel, different shape, and not at time of sunrise, sunset, and no mantras. But otherwise, it's the same substances burned. So you see the big difference also between the effect of control ash and Agnihota ash. So this was by adding Actinoptera ash. Then also the experiment was done by just keeping the plants, the seedlings in Actinoptera atmosphere. And you see the red bars show the treated, means with Actinoptera atmosphere, and the blue bars untreated, no Actinoptera ash. Both shoot length and root length are increased a lot by doing this in the Actinoptera atmosphere. Then there was a lot of research also done in different universities and research institutes on the effect of Actinoptera atmosphere and Actinoptera ash. This was about flowers in a greenhouse, you know, and the stalk length, flower diameter, number of flowers, shelf life, all increased. This experiment was done in Tamil Nadu in Uti, Tamil Nadu Agricultural University. This experiment was done in Agricultural University of Palampur in Himachal Pradesh. Production of lemongrass and Tagetes minuta, quantity increased, oil content increased, and disease resistance was higher. Then there was a series of controlled experiments at Dharwad Agricultural University in Karnataka. 4 MSc theses were produced under the guidance of Dr. Pramod Basarkar. The plants studied were tomatoes, cabbage, soybeans, okra, and they studied yield, pests and diseases, and quality and nutrient content. And in all the parameters, with all the 4 crops, there was an increase. Like here, for example, you see increase in yield from 28% to 180%, depending on what exactly you count. Pests and diseases were controlled in tomatoes, in cabbage, in soybeans, okra, nutrient contents of plants. This is important because studies have shown that there's a decline of nutrient content in our food. And that means also that we may have a lack of nutrients, although we eat a lot. And also the Ayurvedic doctors have confirmed that the medical potency of plants they're using is declining because of pollution. So now, what is the effect of Actinoptera? One result was about vanilla that was done on a Homa farm in Karnataka by the farmer Embryonated Desai in Belgaum area. And he produced vanilla. He gave that to a lab to check the oil content, the vanillin content, and the result was 36% by weight. The lab thought they had made a mistake. They have did the test again, but got the same result. They thought that's not possible because normally they only have 25 to 28%. So that was very extraordinary. Also, these experiments in the Karnataka Dharwad Agricultural University, they checked nutrient content also, and for example found with soybeans 5% increase in protein content, 9.5 increase in oil content. Cabbage Brix value was increased by 39%. Tomatoes ascorbic acid increased 49%. Lycopene 40%. Okra ascorbic acid increase from 0.5 to 34%. Potassium 45 to 98%. Copper, zinc, magnesium iron all also increased. So the nutrient content of plants of the crops increased, which is very important for our health, because our health depends a lot on what we eat. Now, let us look at the effects of organic farming. Means that is the farming where Agricultura and some additional techniques are used, is on biodiversity. I mentioned that at the beginning, that one study a couple of years ago showed that the insect population was reduced by 75% in the last 25 years. Now, what does Agricultura and Entomotherapy help to increase biodiversity? That was one study done by Dr. Shailendra Sharma in Damnot at the Narmada River. And he checked the quantity of invertebrates in the riverbed of the Narmada. And it was shrimps, insecta, Pelecypoda and gastropoda, and it was all added up. And you see the columns. In the middle, this column shows the number of these invertebrates at one homotherapy place in Maheshwar. And left side were 2 places upstream, and at the right side, 2 places downstream. So you see there's a big difference, and the homotherapy place shows much higher biodiversity than the centers, the research stations upstreams, and those downstreams. Similar study was done of butterfly. And here again, you see
out of time now.
there's this echo again.
We are running out of time, I'm afraid, so I think we will be able to continue.
I mean, somebody is talking in between. Can you please stop that? Thank you. So the number of butterflies, again, you see at this homotherapy place in the middle, Maheshwar Goshala, number of butterflies is much higher than upstream and downstream. And here you see a photo showing all the butterflies in that place. Very important also honeybees. And honeybees, that is worldwide a big problem. They are very much suffering from pollution. And we should know that more than 100 crops are pollinated with bees. And it's a big concern if these bees population die, then a lot of these crops will just not grow anymore. And home atmosphere is very conducive for bees, and health, the produced product quality, all improve in the atmosphere of Homa. So to sum up, we can say that Homa organic farming with Agricultura is the basic tool, helps for a sustainable future. This is how we can achieve sustainable development from village level and thus guarantee global food security. And how does this relate to the sustainable development goals, which I mentioned at the beginning? No poverty. Many people can get employed in these small and medium-sized Homa farms. Zero hunger. Enough food can be produced as soil health is improved. And good health and well-being, that is a positive side effect of Homa organic farming. The farmers automatically get a push in the direction of more health. And there are many reports that has happened also. So you see, it's a simple method, very simple method, but with a huge impact in all the different areas of our lives. That's it. Thank you. Thank you very much. For people who know,
Thank you very much, sir.
You
Yes,
can
sir.
contact me with this email address.
Yeah, you are unmuted, ma'am.
So, speaker has talked about the sustainable, a very important tool for the sustainable development on planet Earth, covering entire challenges for the present situation, providing the solution, possible solution, and the positive effect of Agrihotra materials on water, soil, as well as some plant materials. It was a very, very important lecture, and as we have seen in India, the ancient rituals and all the other things that have very, very important impact on at all the level. Whichever is biological energy on the earth, whether it is human, whether it is plant, whether it is say solid and material. Okay, it has an immense impact on all over this. I really, really would have to applaud Dr. Bird because he's talk about that mantra and the pronunciation which were so Indian, and this was really, really applaudable. I'm opening the session for the questions. If anybody has any question, they can ask right now. Or sir has already given the email ID on which you can drop your questions to satisfy your queries. Do you want to ask questions, sir? Yeah, mic is ready.
Yeah, good afternoon, sir. Sir, this the kind of challenge which you are talking is huge in terms of soil which is sodic or acidic, and otherwise also. So, how do you contemplate that we can meet this requirement through this ash contents? This is basically ash is coming from cow dung or from the this thing, branches of burning the branches of a tree like mango, which is which has been subscribed that you should burn this plant specifically. So, is there any difference in the material and the ash produced after that burning material on the all these effects which you are?
Was that the question? For Agnihotra, you only use cow dung, ghee, and some rice. We know that for other homas, different other substances are being burned, like what you mentioned, mango wood. For example, for Soma yaks, you also use cow dung, but you mainly use mango wood. So that's a different thing. And it has beneficial effects, of course, also. But for Agnihotra, it's very simple. We do not need anything. We don't use any Havan Samakri. It is just cow dung, ghee, and rice. And with this simple, very simple, basic Agnihotra fire, we have gotten all the results which I mentioned. Does that answer the question?
The quantity of this thing ash, because earlier even cooking was done on this cow dung fire, but that is what reduced drastically. So I feel that use of cow dung as such after composting and mixing back to the soil will have better this thing, create a better organic material also, and be safe also. It will have better effect than these ash. But that organic material will also be available besides the nutrients or some elements, trace elements which will be there after burning.
Good afternoon, sir. So this is Dr. Sharma, and I have taken those ashes from you to test in my lab. And I also conducted an experiment regarding this ash. I've collected ash from you in 2019 and 2021 I conducted the experiment. But sir, I didn't find any significant difference in the oil content of mustard. So my question is whether the shelf life of that Aviroka or that thing affects the oil content of the nutritional parameter or anti-nutritional parameter of the crop.
Sorry, the sound quality really bad. I didn't understand the question.
The last. The last should be done. Thank you very much. Thank you very much, Dr. Bal. Thank you for joining us in this scientific session. And this lecture is going to be available all the way 24 hours every day on YouTube. So the people, all the audience and delegates will be watching it over and over along with the students. And anybody has queries, they will get back to you on your email ID. Thank you very much for joining us.
Okay, thank you.
Thank you very much, Dr. Berg.
Thank
A
you.
huge round of applause for Dr. Berg. He has joined us from Germany. Sorry, he's in Poland right now.
So
Yes.
this is the end of we are concluding the session, and the delegates will join for the lunch.
We'll start the lunch after a few minutes. We have to honor our chairperson, co-chairperson, and the reporter for both the sessions. Technical session was running here, and a parallel technical session was running in the another hall. And we have to honor, and for that I would request Honorable Director of Hindustan College of Science and Technology, Dr. R.K. Upadhyay sir, and Congress Director, Dr. P.K. Upadhyay sir, to please come forward and honor our chairperson for the session, Professor Kashmira Gohil ma'am. A huge round of applause, please. Next in the sequence, we have the co-chairperson of this technical session, Professor Ravi Upadhyay sir. Professor Ravi Upadhyay sir, co-chairperson of this technical session. Next we have the reporter for the session, Engineer Sandeep Verma sir. I guess sir is not here. No issues. Another technical session was running parallelly to this session in the next hall, and the session was chaired by Dr. A.K.S. Rawat sir. I will request sir to please honor Dr. Rawat sir. Followed by the reporter for the session was Dr. Sushil Kumar Singh sir from Haryana Agriculture University. The session was reported by Dr. Sushil Kumar sir. Also, we have a few awards, as these delegates will be leaving us. They have a train schedule, so I would request sir to please award the following delegates. We have with us Dr. Pushpendra Singh sir from Government PG College, Amarpatan, District Satna, Madhya Pradesh. Sir is being awarded by I.S.G.B.R.D. for Eminent Professor and Scientist Award. A huge round of applause for Dr. Pushpendra Singh. Next we have Dr. Chetna Murania ma'am from Government Nirbhay Singh Patel Science College. Ma'am is being awarded for Best Teaching Award 2022. Dr. Chetna Murania ma'am. Next we have Dr. Priyanka Srivastav ma'am from MGPG College, Gorakhpur. Ma'am is being awarded for Excellence Teaching Award. I would request Professor Kashmira Gohil ma'am and Dr. A.K.S. Rawat sir to please assist in this award. A huge round of applause for ma'am as well. Thank you very much, sir. Heartily congratulations to all the award winners. The next technical session will start sharp at 3 in the same hall. I request all to please gather in the same hall by sharp 3. Also, the lunch will be held at mess of Junior Boys Hostel. All are requested to please move for the lunch. Thank you. Again, a reminder, all are kindly requested to please move for lunch at Junior Mess of Boys Hostel. We'll start with the technical session, next technical session, sharp at 3. I request all to please gather at sharp 3. Please make sure we gather at 3 in the room. Thank you. Sir, thank you. Can we leave, sir?
Sure.
Yeah, thank you, sir. Thank you, sir.
सुनील श्रीवास्तव को नहीं
सुनील श्रीवास्तव को लिंक भेजिए.
हाँ भेज दिया.
ऐसे होगा ये बात? मतलब यहाँ पे नहीं होना है, ऐसे ठीक है, उससे हो जाएगा। शेयर स्क्रीन कहाँ आ रहा है मैम? the line. Yeah, you also come. Okay.
We are going to start with our next technical session, that is technical session 4 of this World Biosciences Congress, BIOSCI-CON 2022. We have with us the chairperson, co-chairperson and the reporter for this session. I would like to invite the chairperson for the session, Professor B.K. Chaturvedi sir, Head Department of Horticulture, Agra University. We have Dr. D.S. Chonkar sir, Department of Agronomy, Agra University, and we have the reporter for the session, Dr. Manoj Pandey sir, from Agra University Department of Agriculture, Chemistry and Soil Science. A huge round of applause for all our chairperson, co-chairperson and the reporter. This session, we have the invited speaker, Dr. Sunil Kumar Srivastava from Department of Microbiology, SSN College, University of Delhi. Sir will be giving his presentation on viral vaccines, past, present and future. Handing over the mic to Honorable Professor Dr. Sunil Kumar Srivastava.
Thank you, Chairperson. I mean, the voice is echoing in. Everybody mute themselves, please.
Yes, sir. Yes, sir. Yes, sir.
Okay. Okay. So the topic that I'm going to speak on today is viral vaccines, past, present, and future. And I have made it very, very simple presentation because I believe this is an educational institution, and there would be many students. So I have brought it to a level so that every student can understand what I am going to talk about. So everybody is hearing about these vaccines these days, particularly about the viral vaccines. Oh, it's not moving. Just a second. Just a minute. Give me. Slides aren't moving. Give me a second, please. Manu? Manu? Slides move नहीं कर रही. How do you do? Okay, I'm sorry for that. There was a paper way back in 2000 that talked about the various kinds of epidemics and pandemics that the world was facing. And as you can see, this paper in Nature that was published by the NIH group, they talked just about the diseases that had the viral diseases and the bacterial diseases which had just emerged from 90s onwards to 2000. So this is just to tell you the magnitude of the problem that we had. We had from time and again so many pandemics that have been affecting the human population, and many deaths, as you see, have been occurring because of them. And apart from the suffering and the economic losses and all those kinds of stuff that has been happening. Then this was a paper that was revised by the NIH group, the Fauci's group, and they came out with another paper in 2017. And then, as you know, in from 2019 onwards, we had another outbreak. So what they tried to capture this. They tried to capture this in 2019, and they found that this was not possible. So they had to employ a graphic artist to do that. So what my message is that we are inundated by so many epidemics from time to time, and we have to have something to deal with these pandemics, and the vaccines are the best bet. So today I'll be talking to you about the vaccines, what had been used by humanity earlier in the earlier times, and what is the situation right now, and what are the projections for the future if we have to protect ourselves from the future pandemics. So So, starting from smallpox, which is a very, very ancient disease, you can see this is a mummy from the Egyptian tombs, and we find that this mummy has smallpox marks on it. So, what I want to convey that smallpox is a very, very ancient disease, and in India we had a practice of what we call as variolation, in which a two-pronged needle used to be used by our rishi munis, and whenever there used to be an outbreak in a village, they used to collect the dry pustules of a person who has already been infected with smallpox, and then they would sterilize the needle and inject this powdery mass into the person's arm. And in the Indian civilization, very, very ancient civilization, this practice was being done, and this was called variolation, and this used to protect the whole village uh, in which the outbreak of smallpox has occurred. This knowledge gradually spread to the Arabic world and to China, and the Chinese started using this. They had devised their own method of snuffing the powder, as you can see this as the picture shows here. And this gradually spread to Turkey, which is in between Asia and Europe. And then Lady Montague, who was the wife of the ambassador of the British to Turkey, spread this message in England. And there was a country physician working in Gloucestershire by the name of Edward Jenner, and he made this observation that the cowmates who used to get a small cowpox were somehow resistant to smallpox outbreak that used to happen in the rural areas. So he was aware of the work of these Indian and the Chinese civilization work, and then Lady Montague's work, who spread the message into Europe. And this is the famous arm of Sarah Names, the cowmaid, from which he collected these dried pustules from cowpox infected mermaids, uh, milkmaids, sorry. And this led to the development of the smallpox as the Western world now knows. And the credit of this goes to Edward Jenner, who in 1796 developed the first vaccine. And this vaccine has been so successful that in 1970s we were able to eradicate the whole of smallpox from this planet Earth, and the WHO declared that a smallpox is dead. Uh, this was a Somalian cook who was the last person to have been infected with smallpox. So this is one success story with which I'm starting. Smallpox, which had been now completely eradicated, and thanks to the procedure of vaccination, which was new to the Western world but was known to Indians long, long back. Then came the chance, an era which we call as the golden era in microbiology, 1850s. And this was a time when Robert Koch in Berlin and Louis Pasteur in Paris, they were doing lots of things in bacteriology. And there was one problem that was confronting Europe or the whole world, was the problem of rabies. Now, rabies, Louis Pasteur tried to do many times because he was a hardcore bacteriologist and tried to see that what is the bacteria that causes it. But he never could get a bacteria because it was a viral disease. However, Louis Pasteur went on to take the rabies saliva, the saliva of a rabid dog, and he inoculated into rabbits. So rabbits were the experimental animals that he selected for rabies. And then, as you can see on the right, he is examining, looking at the spinal cord of the rabbit. And these spinal cords used to dry and then kept on passaging. So people who know vaccinology know there's a term called passaging. in which they keep on inoculating or subculturing the same inoculum continuously for many generations. So Louis Pasteur was doing that, and almost 50 generations of rabies inoculum he had in his lab. Then suddenly one day, what happened is this the boy in the right is Joseph Meister, and he was bitten by a rabid dog, and his mother, Joseph Meister's mother, approached Louis Pasteur and said that, "Sir, you are making a vaccine against rabies, and I want that my Joseph would anyway die because of rabies, because rabies was a death sentence at that time." And she pleaded with Louis Pasteur to try his vaccine on him. Now that was a dilemma for Louis Pasteur, because that vaccine had not been human tested so far. It was in an experimental stage, so it was very difficult. It would be unethical to use a vaccine of that stage on human subject. But she pleaded and she said, "Let it be a success or a failure of science, and I want my son to be the first human subject to do that." So this is the famous painting that we have, Louis Pasteur standing in the right corner, and the vaccine was given, and it was a 14-dose vaccine that had to be given over a period of 14 weeks, and the child's life was saved. He then went on to become the curator of the Pasteur Institute in Paris, Joseph Meister. And this is the famous, as you can see, the newspaper clipping at that time showing that how the angelic Louis Pasteur could save the lives from people who had been bitten by a rabid dog. Okay, as I said, Koch's postulates became very famous because he proved many pathogens that was a germ theory uh, era in which many germs, many bacteria were being discovered, and they were being related or associated as a causative agent or etiological agent for the diseases that they had to be linked to. And for that, Koch made postulates from certain bacteria, including Mycobacterium tuberculosis, in which he proved this. And a lot of interest in vaccination started because we had a handle of growing these microorganisms and then inactivating them or attenuating them, making them non-virulent, and a whole science of vaccinology started. Now, coming to another dreadful disease, that is polio, you are seeing again a clay tablet of an Egyptian priest who is holding the stick. And what this painting shows, that this priest in Egypt was having a paralytic limb, and he was suffering from polio. This is just to indicate that polio was a very, very ancient disease. But the problem with polio was it used to have many epidemics in 50s particularly, but we did not have any vaccine. The problem with polio was it was not a virus that could be grown easily because it's a neurotropic virus. It infects the nervous system. And so it's very difficult to grow this virus in the nerves and get a vaccine. Then there was a John Enders group at Harvard who thought out of the box, and they selected non-neural tissues, and they took the kidney of monkey cells and grew the virus into it, and they succeeded. John Enders, Robbins, and Weller, these three scientists then later on went to get the Nobel Prize in 1954. So Jonas Salk, uh, whom we are seeing on the left, and Albert Sabin on the right, they went on to develop two polio vaccines. And this was a major breakthrough because this would lead finally to almost elimination of polio as we are at right now. We just have polio now in Pakistan and Afghanistan. These are the two countries where we still have polio, but the whole of the world is now free from polio. So Jonas Salk, he went on to develop his vaccine after the culture was available, though he used another cell line which we call HeLa cell lines. And these cell lines proved to be very good from Johns Hopkins. Henrietta Lacks, as you would know, was a black nurse who died of cervical cancer, and her cells were almost immortal and they have become very famous, the HeLa cell lines. And Jonas Salk used those lines, and so did Albert Sabin. So there were two vaccines that were available for polio. One was Salk's vaccine, which was an injectable vaccine, and this was an inactivated vaccine. While Albert Sabin, the vaccine that he developed was an oral vaccine. Now, oral vaccine was very easy to administer. It was a mucosal vaccine, so it would generate the IgA response also, and it would give IgA as well as IgG response, while Salk vaccine would give only IgG response. So this is uh, something which the students of immunology would appreciate. And so this protection with Sabin vaccine was better. And also it was easy to administer. As you can see, this sugar coated vaccine being given to administered by Sabin himself to a girl in just a spoon, on a spoon only. So you don't need a needle or a vaccine, injectable vaccine for that. So as you would recall, in India we had pulse polio program, and this Sabin vaccine was used for that, and that is how this whole vaccine was widely used worldwide, because it was easy to administer, it was easy to store. We just needed refrigerated conditions, not ultra low temperatures, and that is how the success of polio eradication came about in different countries in the world. So India, this is just to show that pulse polio became very active, and we had, now India is free from polio. So 1988, we had uh, 350,000 cases in 125 countries, and 2012 we had 223 cases in 5 countries, and now we are free. India is now completely free, and so are many other countries which could not have imagined that they would be able to eradicate polio. Now, this is just a timeline to show you smallpox. I already talked about rabies and cholera, typhoid. I'm not talking today because these are bacterial diseases. I'm just concentrating on the viral diseases. Polio vaccines, the success stories I've already told you. Now, there were three childhood diseases: measles, mumps, rubella. And it was the genius of this man, Morris Heilman, whom you are seeing down under the chicken pox, this thing. And he was a person who had who has the credit of developing as many as 40 vaccines, and he's called the father of modern vaccinology. So Morris Heilman, who worked for a great period of time in his life with Merck company, he went on to develop these three vaccines against measles, mumps, and rubella. And not only he developed these vaccines in 60s, but what he did, he made a trivalent vaccine combining these together so that it is easy to administer to the children, so they don't have to come for this vaccine again and again. So in one shot, the three vaccines are given, and of course a booster is given later on. So he developed what we call as MMR: measles, mumps, rubella. So the three viral diseases, the childhood diseases, came to be vaccinated by the efforts of Morris Heilman. He also went on to develop a chickenpox vaccine, which was again a childhood disease, and uh, he also developed a vaccine against hepatitis B, as you can see in the left corner of this table, right? So tuberculosis, diphtheria, tetanus, pertussis, and pneumococcal all are bacterial vaccines. So I'm not going to talk about these vaccines today. And then we had a very serious problem, as you would know, uh, after the World War had ended. the Spanish flu outbreak came about, and it was a huge pandemic, almost the size or even bigger than the pandemic that we are facing right now. And the virus was an influenza virus. Now, there were many flu epidemics from time to time, but the problem again was the cultivation or the culturing of this virus. Ernst Goodpasture, who was working at the Vanderbilt, he developed his technique, which we call as chick embryonated egg technique. Now, this was a very elegant technique in which what he did was he inoculated the chorioallantoic membrane of the chick embryonated eggs and could cultivate the virus in that particular tissue. Now, this gave impetus to what we now have, the flu vaccine. And this was the traditional way how people used to grow a flu virus. And thanks to that, as what I'm trying to show you here, that we had experienced a lot of epidemics of flu virus or the influenza virus, but the fatality rate or the toll that these influenza virus pandemics had would have been much higher had we not had the vaccine. So now it is a practice in the US and Europe, and India is also catching up with that now, that every season before the flu season starts, you can take a shot of the flu vaccine and protect yourself against these vaccines. So here we have to remember the mass production of the vaccines of influenza that was possible in growing this in embryonated egg, and that's how we could tweak in every flu season the strains of the viruses. So what happens in every season, in the month of September, the WHO announces that these are the circulating strains, and these companies then use those strains of influenza virus and make a vaccine, and that vaccine becomes very available very fast. And by October, the vaccine is rolled out, and then you can get a shot of the influenza vaccine, flu vaccine, and then you are protected for the entire season. This is especially good for the people who are aged because they are more susceptible to get the flu virus. So this is one another success story of flu vaccine. Influenza virus is a very complicated virus, so it changes very fast. It has both antigenic drift and antigenic shift because, as you can see, it's a multi-stranded 8 strands of single-stranded RNA, and these mutate very fast. And this is what I was trying to tell you: antigenic shift and drift keeps on taking place, and whatever virus is circulating this season, you find another set of strains being circulating in the next year. So every time you have to tweak these and make a vaccine fresh every season. So we had episodes of avian flu, which is again which comes from migratory birds and enters into our domestic bird population. And as you would recall, in 2005 and even before that, we had a lot of uh, outbreaks of avian flu, and many poultry had to be culled, and the entire poultry industry in many countries had to be started afresh. So this is about this a very serious problem, and we are just short of one mutation in avian influenza. So if this mutation occurs, then this disease will be transmitted from human to human. Now it is coming from birds to humans, but we are just one mutation away, and keeping our fingers crossed, we do not know when that will happen because of this high mutating virus, and then maybe we'll have to delve deep into the vaccinology field that and prepare ourselves for this. Then we had the swine flu in 2009, starting in Mexico. And if you see the origin of this virus, as we call it was in the birds, humans, and pigs, swine. So all these three strains of viruses, the avian influenza, the human influenza, and the swine influenza, uh, mixed up in in swine, which is called a blender host, because it's a very good blending host for many viral strains to develop, and as I has shown you here, and that emerged a swine flu outbreak, which I think many of you would know had killed so many people and disrupted the whole. So this is I'm trying to tell you here how this H1N1 strain of swine flu started from avian, human, and mixing with the classical swine, and then the classical swine having this multiple reassorted virus combining with Eurasian swine, and finally you got the swine flu pandemic. In 80s, there was a start of AIDS, though AIDS was there before also, but the first cases that were reported from Los Angeles in the gay people and uh, in prostitutes in Paris, this disease made headlines, and this was it took a very serious turn, and I think we are still fighting with this pandemic. We don't have a vaccine so far, and these three people were the main people who went on to identify the virus, how to grow it, and other things. But still, this anemia is there, and we have not been able to get a vaccine till date. Now, AIDS virus, as just for the student's sake, I'm trying to tell you, it's a retrovirus. It changes its RNA into DNA and then integrates in our body, and then it can remain latent in our human cells for a very long period of time, maybe 10 to 15 years, and then suddenly when the conditions are right, this virus would strike and a person will develop the full AIDS symptoms, and a person can die because of that. It has multiple routes. I don't want to get into that, but there because of these multiple routes and because of the high mutating virus, you find that within a person's blood, you will find so many strains of the virus in the person. If you take the peripheral blood of that person, you will find so many strains of the same virus in a single individual. So that's a big challenge, and that is why we can't keep up with the mutations the virus progresses to have. So it's very, very difficult to make a vaccine because it's all the time a moving target, and it's very difficult to keep track of that and keep pace with the viral evolution. HIV virus is very dangerous virus because it affects your whole immune system. It attacks the CD4 T cells, the dendritic cells, the macrophages. These are some of the host cells, the immune cells that these targets, and this brings down your immunity considerably. And so a person per se doesn't die of HIV, but because of ARC, that is AIDS-related complexes. So there are a lot of opportunistic diseases, the opportunistic pathogens, which a normal person can clear, but a HIV positive person cannot clear because their whole immune system is impacted and is weakened considerably to deal even small infections like Candida, Kaposi sarcoma, and various tuberculosis. So many infections which are normally cleared by the person is not possible in a person who is having HIV. So it's a serious problem. But there is a silver lining, and the silver lining is that we have progressed a lot as far as the antiretroviral therapy is concerned. And what used to be a death sentence for people who got infected with HIV uh, are now being treated with a cocktail of 4 drugs, and these are all antiretroviral protease inhibitors. There are quite a lot of them that are available, though the virus is developing resistance against them. This is one thing that has brought down the cases of HIV infection considerably, though we still don't have a vaccine. Okay, then there was a virus by the name of hepatitis B, and this has also caused pandemics, particularly in the Eastern Asian countries like China, Korea, Japan, and Philippines. and then has spread gradually to the entire world now. And this virus also follows the same route as HIV. This was a very big problem, and the unfortunate thing was that we could not culture this virus. So the vaccine approach, because it needs a culture of the virus, they took another route, and this route was through DNA. So as you would appreciate, there were a lot of developments in molecular biology and advancements in recombinant DNA technology. So they used to, the people associated with hepatitis B virus research work, they isolated the DNA. And what they did was they, as you can see, this is a complex virus, as I have shown in the left side. It has a lot of antigens. AG stands for antigen. And the one in purple, hepatitis B surface antigen, was used, as you can see below, the electron micrographs of viral like particles. And people started taking them from a person, a convalescent plasma, what we call, and this was injected into people, the healthy people, and try to protect them. But this created a lot of other problems, as you would know, and they would get lots of other infections. So the blood banks started screening for HIV and hepatitis B, and a person who was just a convalescent plasma would not qualify to give a vaccine through his plasma. So they started cloning these genes of surface antigen in Saccharomyces cerevisiae, and the first vaccine was developed by Merck company, again by Morris Heilmann, and this technology was later on adopted by GSK, GlaxoSmithKline, and we had a vaccine against hepatitis B virus depending on the hepatitis surface antigen. But the cost was very, very high. A normal low income, middle income group countries could not afford this, and that is where we are proud that India has taken a big lead. Shanta Biotech at that time developed, used this technology and brought hepatitis B surface antigen vaccine as at 1/10 of the cost what Merck and GSK was selling. And this vaccine, it uh, was given to many African countries worldwide. So that is how the genesis of Gavi and all those things started, and COVAX and all those kinds of things started, that where people started thinking of the poor countries also, which did not have this very costly technology available to them. And India has played a major role in this, and we should be really proud of that, how our pharma and vaccine industry has progressed. Now, there was a time when, starting from 1911, that viruses were associated with many cancers. So, if you are developing a vaccine against those viruses which are causing cancer, you are in a way developing a cancer vaccine. So, this was relevant with human papilloma virus, which was found by, isolated by Zurhausen, and who at Heidelberg in Germany, and he went on to prove that this is an oncolytic virus and can cause disease, or which can cause cancer of the cervix in females. And so was hepatitis B. As you know, it can cause hepatocellular carcinoma, liver cancer, and then hepatitis C virus. So there were many viruses that were really being discovered, and it was being found that they can cause cancer. So the good thing was that if we have a vaccine against this, we can prevent those cancers. And then there were many success stories. This is Zurhausen, and this is the cervical cancer that he, and what you are seeing on the bottom is the pap smears, by which we normally used to do the detection of the virus, but now we do it through DNA, HPV DNA. And what happens here is, as you can see, there are particles which are having the nucleic acid. Just see the electron micrographs on the right, and some are without the nucleic acid. So these viruses we call as virus-like particles. So a whole platform of new vaccine was to emerge, where people would use these virus-like particles which don't have the infectious uh, DNA in them in their capsid, and they could be employed because this would act as an immunogen, and the antigen presenting cells would take it and present it to the B cells and the T cells and educate them, and the whole immune system would become active. So this was for the first time done by Sheeler's group, John Sheeler's group at NIH, and they developed this technology and they licensed it to Merck and GSK again, and we had the first vaccine that was developed against human papilloma virus. So not only it would control the viral disease in adolescent people, but also prevent the development of cancer, which usually was the case with chronic hepatitis human papilloma virus. So this is another success story at the bottom, as you are seeing. What I have shown you is the L1 monomer. L1 monomer is one of the capsid protein. We have two structural capsomeres, L1 and L2. So they deployed what we call as L1 capsomere, and this is self-assembling VLP, as you are seeing down around the structure. That is a virus. It is free of the DNA, and then we can easily develop a vaccine against this. Now, coming to the present times, so this was something which I called now the crisis in the present times, and this is mainly because of the zoonotic spillovers. So this anybody now is familiar with what is zoonosis. So viruses jump hosts, and they can jump hosts from bats. This is one of the major reservoirs for many viral diseases, and they can come into our domestic animals like camels and poultry and swine, and then enter the human being. So this is called zoonotic spillovers for the students. So what is happening is finally it is entering into a human host. Please remember, human is not the host for this particular virus. So it takes time for this virus to adapt in the human host. Now, the burning example of this is SARS-CoV-2, or for that matter, SARS we had experienced in 2002, and the MERS. So they have come from bat. They went on in the palm civet cat in SARS, which occurred in 2002, and in camels, as we saw in MERS. And then from these two animals, they jumped into human beings. Now, thinking from the virus point of view, human is not a host, so it takes time for these viruses to adapt in the human host. And till the time they are not able to adapt, they will cause a lot of suffering and even death, which the virus actually doesn't want to kill the host. So this is what all we have been experiencing, let's say from 2019 onwards in case of SARS-CoV-2, and now gradually the virus is seem to have adapted in the human host, and that is why you are seeing less suffering, less deaths. So gradually this virus will adapt and we'll have to live with this virus. Okay, so bats, as I said, are the universal reservoir of viruses. This is the different species of bats. I don't have time to go into that, but this is also very interesting. And India is, the Northeast is having a variety of bats, different uh, diversity of bats altogether, and this has to be studied properly because these are some of the reservoirs where many new viruses we can expect to come in to the human population. Coming of this bat transmission, Ebola is one very serious, very dreadful disease that Africa has been experiencing from time to time. In 1976, this virus was discovered by Peter Poy and a Belgian scientist, and then it has since then caused many epidemics in Democratic Republic of Congo and has spread to many other African countries. And this is a very serious disease. This virus, as you can see, the shape of the virus on the corner, and this virus can infect all your human body organs. It is a systemic virus, and it goes and causes hemorrhage, and any part of your body can start bleeding, maybe your eyes, nose, ear, and through mouth also the blood starts spilling because the virus causes a lot of hemorrhage, and it used to be a death sentence uh, whenever Ebola outbreak used to take place. Still, it causes a great deal of fatality. This is the virus. It's a very complex virus. It's an RNA containing virus. This is a shape of this virus. This has lipid envelope around it. And if you see the outbreaks, as I was trying to tell you, since 76, we have been experiencing a lot of uh, epidemics restricted to Africa. In 2014, there was a major outbreak, as I have shown by the size of the circle. This was a major outbreak, and
Please sum up. We have 4 5 more speakers in this session.
okay, I'm just trying to end. But please mute yourselves as well because it's echoing. So Ebola was a very dreadful disease, and the problem was the people who were engaged with the ceremony of these last rites. They also used to get infected. So the PPE started from there actually. The use of PPE, as you can see, these people then started. So this is the bat, the fruit bat, which harbors the virus. So Charles Anson, he is a retired scientist now at the University of Arizona, and he started a new technology which wherein you would grow the viruses in plants. And we had some beautiful vaccines being developed by his lab, and this was ZMAP. Z is for Ebola, and MA is for monoclonal antibodies. So, as you know, in 2014, there was an outbreak in Western Africa, and many people were dying. And Anzen again tested his vaccine, which was again at an experimental stage. It was just being challenged. The monkeys were being challenged, and at that stage, through compassionate grounds, the U.S. government asked him to make this vaccine available, and this vaccine was used uh, in Ebola outbreak because these two people who were serving in Ebola from NIH, Dr. Ken Brentley and this nurse, Nancy Wright-Bull, they got infected with Ebola and their life was in danger. And for the first time, these two persons were tested by a plant-based monoclonal antibody, which had not gone even into human trials. So this is for the first time in the history this happened, and the lives of these two people were saved thanks to Barack Obama's compassionate ground that this vaccine was used. And here you are seeing Brentley speaking after his return from West Africa. So this is so now coming to relating to the present times, we can use whole inactivated vaccine. I'm trying to correlate with SARS now. The one that was used by Bharat Biotech in India, live attenuated vaccine, and then live attenuated was not used in SARS. Then we had other vaccines that were used by this. So coronaviruses are a very big group of viruses. I don't have time to go into that, but we had epidemics of SARS, MERS, and now we have the SARS-CoV-2. So what people did, the red area, the spike region, they isolated its RNA and transformed into cDNA, and this was then used against various strains of viruses that we keep on thinking. So again, SARS-CoV-2 has been a moving target. It keeps on changing very fast, and it's very difficult to really aim at it because it keeps changing its antigen. So so this is, I just wanted to discuss about Delta and other things. Sorry, I don't have time, Omicron and things. But so it's a moving target altogether. But what I just want to tell you, through this epidemic, we have gained a lot as far as vaccinology is concerned. In record time, we had made a vaccine in less than an year's time, and this was possible. This is the Oxford group, the ChEDOX group, about which I have just discussed the methodology that they use. They used a recombinant vaccine in adenovirus, and this is what we have in Covishield. And then this was the spike protein, and this was the spike protein vaccine that has been used by Novavax. And this is the famous mRNA vaccine, which was being earlier tried for cancer, but nobody had tried it ever in any human subject. So once this 2019 this outbreak occurred in Wuhan, the in January the Chinese CDC released this spike protein data, and this people then would download from the cloud. Many companies started that in January itself, and as you can see here, the messenger RNA sequence was then uh, elucidated, and everything then was manufactured synthetically. Please remember that word. Now we have moved to an era where we are using a synthetic vaccine. We don't have to have the virus per se. So we here, as I'm trying to show you, there you have a purified mRNA, and that then you can formulate it in nanoparticles, lipid nanoparticles. You tweak a few RNA. I don't have the time to go into that, but this is a technology that is available, and for many other viruses also this technology came to be used about. And plant-based vaccines, just a word on that, because this is a very good technology wherein you can develop your virus like particles, maintain them, culture them, multiply them in plants, because it's easy to grow things in plants. This is all the methodology. I don't have the time, sorry for that. But now we have a vaccine that is available against SARS-CoV-2 also, produced in Nicotiana benthamiana. This has been done in a few countries like Canada, and the company is Medicago. You can just Google it and see it. And this is one of the success stories that we have. And this platform is very, very good, because for a country like India, where there is no dearth of sunlight, we can easily produce this. This would be a low cost vaccine. This could be an oral vaccine. For that, you don't need needles even. And this could also circumvent the necessity of very, very cold, ultra cold temperatures, which is a major stumbling block for many of the low middle income countries. So I think I'll end here. This is the vaccine production for SARS-CoV-2. And thank you so much for your attention. So I would stop here.
Thank you, sir. We have with us Dr. Srinivasa, sir. Very nice presentation about vaccine and viral diseases and pandemic whatsoever going in last 2-3 years, and all about for those interested in microbiology and in related areas. So thanks, sir. Now topic open for the discussion. If any of us among the audience, online or offline, have any query or question,
I can pass on my Arun has my email. He has been my student.
Okay, sir.
So
So
you're
here
free
we,
anytime to contact me through him or
okay, sir. Okay. So we have a sort of time here. Many speaker in this session. So thanks, a lot of thanks, sir, for your kind presentation. A very nice presentation here in this session. And now I request to for the next speaker of this session.
The next speaker for this technical session is Professor Shurveer Singh from Department of Biotechnology, JRA University, Mathura.
रिकॉर्ड कैसे करेंगे रिकॉर्ड ये कैसे होगा तो उससे रिकॉर्ड कैसे होगा ये निकाल दो भैया शुमन ये निकाल दो आगे करते हैं
So next speaker in this session, Dr. Shurveer Singh, he is Professor of Biotechnology in GLA, Mathura. Sir, please start your presentation.
Thank you so much. So I am at the outset very thankful to Dr. Upadhyay and Hindustan College for inviting me and permitting me to present my work. Actually, I passed my best time of the life right here, just 5 kilometers away from Hindustan College. This college came before my eyes. I have seen the development from the day one, but I rarely had the opportunity to come into the institution because our hands were full with the disease I am going to talk to you. When I joined this Center Institute for Research on Goats, it was established 12th July 1979 as an institute. It was earlier a center of I.G.B.R.D, where earlier it was a UP government bull rearing farm. Then it was handed over to I.G.B.R.D for as a goat development center, and they were sending their culled animals to this place, being a remote area. So in 1984, when I joined this institute, there was nothing like a microbiology lab. We had a health division. There were goats, which we had to take care of their health for different diseases. As you know, the goat is an Indian or an animal of the poor. It's poor man's cow. So there was very little literature available on goats. Talk about their breeds, talk about their nutrition, reproduction, or health care. So most of the thing, even in our veterinary science, when we are taught about this different livestock, the horse is the typical species which used to be taught. Then comes the cattle and buffaloes. Goats and sheep are always skipped over, and something about a pig, dog, and some poultry. So everything was new to me, and I never joined veterinary science to be a doctor. I wanted to be a scientist, that's why I joined ARS services. I never applied for any veterinary officer's job. But then there was no laboratory here, so it was a difficult time for me for next 16 years. I was not having any lab. Only in 2000, after 84, joining here in 84, I got a lab, vacant lab, just like a room. And then we slowly developed into it, and all my achievements are there from 2000 to 2018. And the disease, which was also again pushed over during my college time, was never taught in detail, like para-TB. I had to spend entire my life on this, and this is a disease which infects both domestic as well as wildlife, ruminants. So this is my entire, you can say, career, or it's my current bhumi. I studied in Mathura Veterinary College 6 years, 77 to 83, doing my graduation. and after applying for the job, I got this posting here on All India Services. And after retiring from here in June in 2018, I was asked by GLA to join them and head the Department of Biotechnology. So this is my institute, the building which came in 1999 and we entered into 2000. So if you see this microbacterial diseases, you have heard about the vaccine on viruses, which are very tiny molecules, but it is again a big challenge to have bacterial vaccines because they are big bacilli, they have a number of antigens, so most of the time the technologies of your mRNA, subunit vaccine, recombinant vaccine, or for that matter, other type of DNA vaccines, they don't work here. And since I started my career in a very primitive lab, but I was lucky to have a guru. And when Dr. Anubhuti told about Randhawa sir, I was all the time looking forward to meet him because it is the guru which makes all the difference. It is not the laboratory, it is not the this you can say the equipment which make the difference. So it is the guru. So I had a head of department pathologist, Dr. Neil Singh, who introduced me to this disease. And when the first day I joined this institute, I was asked to look after the treatment of the goats. Sir, when I went to this one of the farm of Burberry goats, 30 animals were standing in diarrhea. So the compounder was giving sulfonylurea, tricyclic and giving. I said, what is this? Sir, it was Jones disease. तो क्या करना है? तो बोले सर इसको देते हैं हम, फिर वो डायरिया रुक जाता है, ऐसे चलता है. Then the sir told me that let us start screening them microscopically. Start taking fecal samples. Sir, from that day we started screening the flock for this disease. And if you see, sir, our livestock, India is blessed with the best wealth you can consider in terms of the livestock, the breeds, nowhere unfeathered, and the population is very huge. We have more than 101 recognized breeds of different these 4 major livestock species. And if you see, sir, I would like to have, sir, your attention that this is one disease which is affecting the population dynamics of this domestic livestock. There is cattle or cow, buffaloes, sheep, and goats. You can kill or cull or slaughter buffaloes, sheep, and goat. So their population is continuously increasing. If you see that, but the population of the cows now, farmer is no longer interested in keeping cows because when it is off production, there is zero salvage value. Either he has to leave it on the road or somewhere. But other animals, still he can sell it, and that is sold for this slaughtering and for the meat production. So this is a disease you see. The major this perspective of this disease, it is zoonotic, it is a production disease, endemic, chronic, incurable, multiple species disease, silent killer, globally prevalent, impacts the global economy, trade restrictions, public health significance, present in wildlife, and so and so forth. So the pathogen which causes this disease is called Microbacterium avians paratuberculosis. It is this father of this bacilli is the Microbacterium avian, which is a bird. This thing, paratibi. Then the disease is chronic, I said. That slowly animals goes on becoming weak, maybe due to persistent diarrhea, but now the symptomatology has changed in the last 30 years. I am seeing that animals are weakening without diarrhea. So it's a progressive disease, progressive waste loss, wasting and death. As I told you, it's a wider source range, primarily the domestic or the wild ruminants, then other animals, primates, human beings. Then actually this disease causes negative energy balance. As I told you, it's a production disease. So whatever animal is eating, digesting is not going into the body. Through diarrhea, it is losing the protein which it is digesting. It is not going into this system either for production or for the growth. Then it damages the immune system and make the animal weak, this immune system weak. They become susceptible to other infections. And the most important thing is that this bacilli escapes pasteurization temperature. So whatever milk products you are eating, my children or my students who like ice cream, it's very dangerous for your health. You can eat ice cream, but once in a while. If you are eating more, you are likely to get this infection. And this is again very difficult to diagnose, very again difficult to cure. So then there are a number of human diseases, more than 22, where it has been listed as the cause of this disease. Most of them are of autoimmune nature. It's a global disease, virus associated, and transmission is both from parents to offspring or the next generation, during pregnancy, through semen, or through milk and cholesterol feeding, as well as from the horizontally from the environment, from the water and other sources. Sir, when I joined this institute, the animals were looking like this, with the best goat breed of the country we are proud to have, and it is found only in a small block of Itawa called Chakarnagar. From there we brought the animals to conserve, because we are solely using, that's the best milk producing breed of the Asia, called Jamnapari. We had a flock of Jamnapari, herd for Jamnapari. Despite sitting over there from 84, this is a picture in 2000, killing the infected animals. Animals reached to this stage because disease was continuously transmitting. The bacteria was gaining in pathogenicity, and they were so much infected that looking like hidebound. Despite they were getting the best of nutrition, which I can't imagine for myself, the kind of nutrition is giving to these animals, considering as a very precious breed of the country. So this is the impact of the disease. From intestine, as I told you, it may cause diarrhea, it may not cause diarrhea. Then in intestine, in the large intestine, it forms corrugations. These I have never seen before in this institute, despite I have conducted more than 5,000 postmortem. Because any goat died, we have to put it to postmortem, we have to see. So all the animals, they die before reaching to this stage. This is a slide from a goat died in Odisha. Then you can see this internal lining of the mucosal lining of the intestine, where this bacilli grows, they burst, then infects the new cells, and this is how this entire lining is removed. From intestine, it goes to lymph nodes. In lymph nodes, the germinal centers are destroyed, and there is no antibody forming. So animals, they are crippled. So this is a picture of cows. That's the healthy cow, and you can see the other, this weak cow. It's a beautiful breed of Haryana. This is a typical case of Yemeni disease. If you see such kind of animal on the road, they are affected with disease. There's nothing, you don't need any test. And now see their cows. What kind of parents or mother or the cow or the buffalo they will become. Their mother was infected. This is a form from Palampur, veterinary college, Jersey breed. So these production losses which I have narrated over there, they are not considered as losses in the country. In India, we consider if an animal dies, that's the only loss. But majority of our animals are low producing because of such infections. So as I told you, it's not an Indian disease, it's a global. More studies have been done in the Western world. And here is the data, all published data, and you can see in the United States now it's more than 72% of their dairy cattle herd are infected with Jones disease, 72%. So you can imagine the condition in India. So outside developed country, there is a very little record of this disease. This is why we are not prioritized the disease for the control, though it is number one disease to be taken care of, both from the animal production perspective as well as human health, to prevent the human infection. So see the losses they have calculated on this basis the maximum resources on they have spent for research on this disease. Every country, because they have the data with them. In India, we have some little information, and you see, they say the goat meat is lean. Why goat meat is preferred in India? Being a tropical country, goat meat is lean, that is why. Less fat, but goat meat is lean because of Yemeni disease. If goat doesn't have the Yemeni disease, it will have same fat as sheep. Sheep is preferred in the TNK. All these Himalayan regions where the cold this thing is there. So there are very few studies where we have calculated the losses, because I will not, because there is a shortage of time. So these 4 tests over a period of time we standardized and we used. Until now they are being used there. We started with microscopy. Culture is very difficult. After leprosy, this is the most difficult bacteria to be cultured. It takes 100 days, more than 100 days, for a small colonies to appear. That too in a media where we have to provide a growth promoter, which costs now more than 10,000 per 2 milligram vial, wherein we can make only 1 liter of media. So these are very old colonies. These are more than 4 or 5 year colonies.
So the generation time must be very large.
Yeah, it must be very large, sir. Yeah. And then we developed PCR after 2000, and then we developed ELISA. We started developing ELISA with the help of most of the earlier studies. We took the help of Central Leprosy Institute in Agra, because they were having all the facilities to study leprosy or the tuberculosis. So we took their help in making our antigen, initially harvesting it because we didn't have the culture, confirming this bacteria as a para-TB. They had the facility of this radioactive this thing. So making up this radioactive probe, we could confirm this disease. So we are very thankful to this the team of Dr. Sen Gupta, Dr. Katoch and others. They helped with large-heartedly, and they helped in our studies. So these are the tests we developed. The 4 tests I told you, they are routine. And then there are other tests, depending on your need and this requirement, you can use upon the test either the antigen detection test or the antibody detection test. Because in a chronic infection, you have to have more than 1 test. The problem with chronic infection is the detection and the confirmation. And this is the picture from the stool sample, the one you can get it in your sputum if you are infected with the open case of tuberculosis. So you get it in fecal material here. It's a pure colony of this para-TB. And this is the ELISA kit which we developed, we have commercialized, and we are ready to sell it to any company for the screening of the animals. And here we are getting this picture after applying this test. The samples in positive and strong positive are taken as positive, and others are taken as negative. Now this is the 32 years study. The samples which we received here, there were around 26,000 samples, fecal sample, milk sample, blood samples, so on and so forth, from different places, different livestock species, and after screening them with one or these tests, you can imagine that from 11.4%, the infection has become double, more than double. Because we are not doing anything to control this infection. And this data depends on the screening of 26,000 these samples which we tested. So this is our typical PCR test where we can identify specifically that it is nothing but para-TB. And this is the biotype. It's an Indian biotype, or Indian bison type, which we have first time discovered that there was only one report of this bison type from the US, only one report, but our bison type is different. So we are now telling it as an Indian bison type. And we have registered it with the NCBI, and you can see majority of the samples which came were coming to be this Indian bison type. We very few times we got cattle type. Sheep type we didn't discover here in India. In wildlife, you can imagine, so much is the infection rate. Then in your environment, surface water, soil water, municipal tap water, these are studies from abroad, it's not from India. Domestic water supplies, this is the ones we have done in soil and water in this region. There we could get this infection in our campus, the soil was infected. Then there was lesser infection in the soil from Ottawa and other places. Then the waters, because we do I.S.G.B.R.D. So somebody sent me this Yemeni water, which was highly contaminated with the other bacteria plus this paraTB also. So you can imagine the level of infection, and these are the reports of the in public health and the milk and milk products. These are all Western reports, published reports. We are having this in individual milk sample, raw milk samples, curd, hard cheese, semi-hard cheese, by culturing, so it is present live in all these milk and milk products. These are all Western studies. There are now some of our Indian studies. Then we started to see that how it is moving from animals to human being or next generation. So then but in beef cattle, the infection rate is little less. It is majority of the milk animals they are more susceptible to this infection. Now this is our Indian studies. Again, we see a lot of contamination of the different milk and milk products which we purchase: paneer, pasteurized milk. Then we had the milk products, and now you see the commercial milk, the milk powder which you are giving to your babies in the home. These are 15 brands. This is not 15 samples like Amul. This is not 15 samples from Amul. There are 15 brands which are available in the market. Ice cream, 16 brands, cheese, curd, lid microscopy, PCR, ELISA. So all are contaminated because we are not doing anything to control them. You see how much is the shedding. You can see how much is the quantity of the bacteria present in this milk and milk products. This is over a drop of milk. This is the kind of infection that is coming. It's from a buffalo. Over a drop of milk, this much is the load of bacilli. And even if you are boiling it, they are causing allergies, different kind of allergies which we are talking. The gluten allergy, the something other, they are because of this. It causes this allergic rhinitis, so and so forth. So this is a culture. This is very difficult to culture these colonies from these milk samples. Then again, milk samples. Then it is not one test, because you are always questioned that you are giving such a high prevalence. What's the basis? We are always questioned. They don't believe this. India is the first country where a person working for last 40 years is questioned about this data. When I presented this data in all the world forums, nobody questioned me. Rather, they follow me. They look what he is telling. But here, first thing is the administrator or even our fellow scientists, they question. So that is why we have to use more than one test. And you see, on a single test, there are very few samples positive. All are present on multiple tests. So that means they confirm they are having. So this is the human link of this disease, proven by Crohn's. Similarly, this inflammatory bowel disease which we have, or the Crohn's disease, have a similar symptoms, which was told by Dr. Crohn's, and after him is known as Crohn's disease. When Sir was telling yesterday CD, I was confused that he's talking about Crohn's disease. So they have almost similar symptoms, similar picture which I have shown you for the intestine of the human being. Sir, this is the data from the Mathura. We collected from different pathology labs, the material they were throwing the stool sample after testing or this blood or the serum. But we just know what purpose it was submitted, but result we never knew. So the human being, they are also known to have a cell wall deficient form. There is a debate. That is why it has not been so far told as a zoonotic disease. But in human being in India, we had similar cell wall, bacteria, and that was also to the presence was quite high, 7.1%, and even more than that. So now this is the samples, the serum samples. I collected more than 28,000, but we got exhausted after screening because similar results were coming. After screening 19,775 samples, 34.3% population was having active disease. Disease was active in that. So this is the level of prevalence of this infection which is going through milk and other milk products. And you see the diseases, the human diseases where it has been associated. This conclude.
Sir, it means that whenever immune system is weakened, okay, then this pathogen might be.
That is also there, but we get it from different means and it sleeps over. It waits for the right opportune time to strike you. So just I will have 5 minutes, sir, from the chairman, sir. To control, we were using test and cull because treatment is not possible. If you use a drug for 7-8 months, it will run over the cost of the animal. So treatment was out of question. The test and cull was the best policy. Since goat was nothing to bar from culling a goat, we are culling these goats, but it has not yielded any positive response. We couldn't reduce the load of this bacilli in the herd or the environment. Then we developed a vaccine in 2004, and we developed a our own vaccine. This is why I have given it to title my own name, homegrown vaccine. Such an environment I have told you where I studied, and in that environment I made this vaccine. It was tested and it has salient features that it was both prophylactic as well as therapeutic. It can cure also infected animals. And these are the parameters on which this vaccine was tested. Physical traits, reproductive parameters, production parameters, immunological parameters, the status of this shedding, which is very important. And these were the number of trials which we did to exhibit that this vaccine works. And you can see here, you will appreciate that first one is where I use my vaccine, second one is the good air vaccine imported, and this is a control. So whenever I did postmortem, animals, these organs used to come before me, despite best of feeding. Only after vaccination you see the fat layer generated to the tune you get in sheep. And lymph nodes, you can see the enlargement. Though this vaccine gave some positive results, imported one, but it couldn't prevent the pathogenesis of the disease. So then we did challenge trial, and then we did in this flock. And you see this, you can see the chain. The parameters before vaccinations and parameters after more than double, more than 3 times, 4 times, 5 times improvement. One vaccine can change this scenario. It's a wonder vaccine. If this country applies today, not only we can boost the productivity of animals, we can use these cows which are moving on the road and prevent the human infection. Half of the clinics will be shut down. We never consider why there is so much of hospitalization. What has happened suddenly? So this is there. Even sir, during COVID, if you see the mortality, the Western world had a more mortality despite they look very healthy because their immune system is compromised. They depend heavily on milk and milk products from 1960s. Since in our country also metropolitans, you are eating this milk and milk products. Here also in India, the mortality was higher in Delhi, Bombay, and other metropolitan cities. In rural areas, there was no problem. So basically, it is your immune system. If your immune system is compromised, no vaccine will work. As you say, na, कि वो सब vaccine लगाया कुछ नहीं हुआ. But if your immune system is
compromised, then how you are going to have this the effect of the vaccine? So then we tested, and now you see the change. Just after 2 months, this is a picture of these animals. The other data I have given you.
Please conclude, sir.
So, using this vaccine, you can not only boost your livestock productivity. Now see these animals, first time when I gave vaccination, how they improved after 7 months. And you see in Jaffrana also the milk production after vaccination, 3.85 liter per day. And this is in calf, which was getting best of feeding, but was not growing, coming negative in the test. But when given a specific vaccine, it bloomed. So you can see the rampantness. This the cow is on the road. After giving vaccine in 1 year time, how much it has changed. So this is the vaccine which we developed. It has been licensed by Drug Control of India. This is one of the very few veterinary vaccines which has been licensed, given approved by Drug Control General of India, was released by the government of India, CSIR, and was also awarded this thing by NRDC for producing this vaccine. So this is now one of the best semen technologies that has ever come out of the CSIR system. But unfortunately, there are a number of people who came and worked, and I remain continuously with disease. Still I am working, I am having now 6 projects on this disease, basis of the leads I have developed. I have more leads, so I have to thank them all. I cannot say simply for each of them. Is Dr. Naim Singh, who introduced me and who was with me till 2004. And Dr. Nirmal Ganguly, he, for the final vaccine which I showed you with the BioVac, he was the chairperson who was evaluating this vaccine. Dr. Katoj helped, and Dr. Singh Gupta in our initial days. Thank you so much, sir.
Thank you, sir.
Sir, is there any possibility that this can work in humans also?
Yes, sir. Why not? We can have similar vaccine for TB.
Yeah.
But already there are problems with this disease. It is a simple kill vaccine. If it's not going to do any benefit, what it will do, a 1 ml of oil, if you put it below the skin of a cow or a goat.
Sir, I'm sure.
But still there is no, and there is so much daily PM is asking, CM is asking what to do with this cow, what to do with this, these farmers are troubled. But here is a solution, but since the solution has come from a very small place, that is the roadblock is because of the place. Had it been developed in Bombay, had it been developed in I.I.G.B.R.D. Delhi, it would have been received like anything. Thank you so much.
Thank you, sir. We have 2-3 more speakers, sir. Very nice presentation and overall on the livestock and the para TV. So many things discussed by Dr. Surya sir, Professor Head Biotechnology. Lot of thanks, sir, for your contribution in this livestock field, and congratulations to you for developing this nice vaccine for livestock people. Thank you, sir.
Thank you very much, sir. Now we have from MD Panchmani, Professor Ravi Upadhyay, sir.
So please keep in mind time duration. We have some more speakers.
Yes, not more than 5 minutes.
Yes, sir. Brief is always best.
But good afternoon, everybody. Please, I want to thank Dr. Polio Upadhyay and apologize for all the guest speakers because I was leaving for MP today immediately, so I have to sum up in 5 minutes and I have taken the time of yours. तो मैं हिंदी में बोलूँ या अंग्रेजी में बोलूँ? हिंदी में चल जाएगा? चल जाएगा बढ़िया। तो मैं शुद्ध हिंदी में बोलूँगा। तो पचमढ़ी से आया हूँ भैया, मैं बायोडायवर्सिटी में काम करता हूँ, प्रोफेसर हूँ और गवर्नमेंट ऑफ मध्य प्रदेश का मैं एडवाइजर भी हूँ बायोडायवर्सिटी के ऊपर। हम लोग पीपुल्स बायोडायवर्सिटी रजिस्टर बना रहे हैं मध्य प्रदेश में, और ऐसे पीपुल्स बायोडायवर्सिटी रजिस्टर हर गांव में, हर ग्राम पंचायत में और कहा जाए हर जनपद में बनेगा पूरे देश में। क्योंकि गवर्नमेंट ऑफ इंडिया बायोसिक्योरिटी एक्ट 2002 के अंतर्गत ये सब काम होना है, तो वो सब काम वो बाद में करेंगे, कभी कभी कर लेंगे, मैंने छोड़ दिया है। आपको पचमढ़ी दर्शन कराया था, कभी आना हो पचमढ़ी, आप मोस्ट वेलकम। इट इज वन ऑफ द ब्यूटीफुल प्लेस ऑफ मध्य प्रदेश। और क्योंकि पचमढ़ी जो ऐसा फॉरेस्ट है जो वर्ल्ड हेरिटेज साइट भी है इंडिया में। इट इज आल्सो ए बायोस्फीयर रिजर्व एज वेल एज वर्ल्ड हेरिटेज साइट एंड सतपुड़ा टाइगर रिजर्व, तीनों एक साथ है, ऐसा किसी भी फॉरेस्ट में इंडिया में नहीं। तो चलिए दर्शन करते हैं, इसके बारे में क्या क्या है इसमें। आप कुछ नहीं करते। खास बात इसमें यह है कि 1974 में बना था, इसके ऊपर आप देखते जाना फटाफट, मैं आपको दिखा देता हूँ। ये कैप्टन फोर्सेस से शुरू होती है बात। एक तात्या टोपे का नाम सुना होगा आपने, 1857 की लड़ाई में। उस समय तात्या टोपे को फॉलो करते हुए कैप्टन फोर्सेस पचमढ़ी चढ़ गए थे, क्योंकि तात्या टोपे वहाँ पे छोटे मोटे मारा गया था, और उनको ढूंढते ढूंढते पचमढ़ी पहुंचे, और पचमढ़ी उनको बड़ा अच्छा लगा। वहाँ पे उन्होंने फर्स्ट अपनी वहाँ पे जाकर कब्जा कर लिया, और वहाँ पर उन्होंने अपना बाइसन लॉज वहाँ स्टार्ट की। कभी आप जाए तो देखें। लेकिन पचमढ़ी का इतिहास बहुत प्रागैतिहासिक समय से पचमढ़ी है, क्योंकि वहाँ पर पचमढ़ी का नाम जो है पांडवों से है। 5 मढ़ियाँ हैं, 5 गुफाएं हैं वहाँ पर, उसका नाम पचमढ़ी है। ये पूरा एरिया है, उसका कितना कितना क्या क्या एरिया है हम टाइम वेस्ट नहीं करते। वहाँ बहुत बढ़िया झरने, आपको 12 महीने बहता हुआ झरना मिलेगा, शुद्ध पानी का झरना। जंगल जो है हमारे यहाँ 2 तरह के जंगल है जो एक साथ पचमढ़ी मिलते हैं, दैट इज साल फॉरेस्ट एज वेल एज टीक फॉरेस्ट। अब सोशल एट्रीब्यूट्स बहुत सारे हैं, जो हमारे पास करीब 9 तरह के ट्राइबल ग्रुप्स हैं, वो बहुत तरह के ट्राइबल मेडिसिन करते हैं, बहुत सारी जड़ी बूटियां बेचते हैं। 1500 से ज्यादा जड़ी बूटियां वहाँ पचमढ़ी बिकती है सर। और ये वहाँ पर जो रॉक शेल्टर्स और डॉग आर्ट्स है, ग्रीनबेल्ट का आपने नाम सुना हो, और पचमढ़ी में भी बहुत सारे रॉक शेल्टर्स पूरा टाइगर रिजर्व में। तो वहाँ पर आपको देख सकते हैं 10,000 से लेकर के 50,000 साल पुराने रॉक शेल्टर से सही ये प्रमाण मिलता है कि ह्यूमन सिविलाइजेशन उस क्षेत्र में 50,000 साल पहले भी थी, और उनके हाथ में धनुष मार है। सबसे बड़ी बात यह है वो कोई स्टोन पेल्टर्स नहीं है, जो पत्थर फेंकते हो, या उसको हम कह सकते हैं आदि मानव नहीं है। उनके हाथ में औजार है, तलवार है, ढाले आप देख सकते हैं। ये भी उसके जो लिस्ट है उसको दिखा देता हूँ। एल्गी है, वहाँ पर 3 रेयर एल्गी मिलती है। एक रेड एल्गा मिलता है, इसको बोलते हैं Verticospermum, इट इज ओनली फ्रेश वाटर एल्गा जो फाउंड इन इंडिया, और वो पचमढ़ी मिलता है। इसके बाद तीनों एल्गा जो है केवल पचमढ़ी में मिलते हैं। फिर इट इज a paradise of bryophytes. You can see about more than 125 bryophytes in this region, and 150 pterophytes can be seen. पचमढ़ी से रावत सर बैठे, रावत सर ने चाहे किताब भी लिखी थी, मुझसे कभी पेपर मांगा था, मैं दे ही पाया, क्षमा चाहूँगा टेरोफाइट्स के ऊपर। तो यहाँ पर हमने जो काम किया है एंटी कैंसर प्लांट्स के ऊपर, तो एंटी कैंसर प्लांट्स में स्लैजिनेला बायोक्रिस और इसका हमारे 3 पेपर पब्लिश है, जो कि नेक्स्ट ब्रिटिश जर्नल ऑफ न्यूट्रिशन से पब्लिश है, जो बहुत अच्छा रिजल्ट मिला है एंटी कैंसर एंटी इन्फ्लेमेटरी प्लांट के ऊपर। उसके बारे में कभी फिर बात करेंगे। स्लैजिनेला बायोक्रिस और साइटिया जाइगेंशिया, ये साइटिया जाइगेंशिया, अल्सोफिला, और वहाँ पर एंडेमिक स्पीशीज मिलती है अल्सोफिला बालकृष्ण नायक, जो इज ओनली फाउंड इन पचमढ़ी थ्रू आउट द वर्ल्ड, कहीं नहीं मिलती, केवल पचमढ़ी मिलती है। फिर सैलोटम देखा दुनिया में बहुत पूरे भारत से लोग देखते हैं, आते पचमढ़ी में आइसोलेटेड सैलोटम, ऑस्मुंडा, ऑफियोब्लॉसम भी करीब 10-12 स्पीशीज पचमढ़ी में आप देखने को मिलेंगी, जो आपको बहुत जगह रेयर है। ये सारे टेरोफाइट्स दे आर एंडेंजर्ड, which you can see in abundance in पचमढ़ी। यहाँ पर आप देख सकते हैं इंसेक्टिवर्स प्लांट्स, बहुत तरह के इंसेक्टिवर्स प्लांट्स आप देख सकते हैं, जो कीट भक्षी पौधे कहलाते हैं, जो पचमढ़ी में आप देखते हैं। इसमें ड्रॉसरा है, ड्रॉसरा की 3-4 स्पीशीज मिलती है, रोटिकुलेरिया की 3-4 स्पीशीज मिलती है। फिर औषधि पौधों की तो भरमार, मैंने आपको पहले ही कह दिया, बहुत सारे जो आपने देखा होगा बीच में दारू हल्दी, बड़ा अच्छा नाम है दारू और हल्दी। दारू हल्दी इसलिए कहते हैं, दो कारणों से दारू हल्दी कहा जाता है, क्योंकि दारू, एक तो दारू जो बनाई जाती है उसमें इसको डाला जाता है, ताकि उसमें दारू को कलर नहीं होता, उसका जो गोल्डन कलर आता है वो दारू हल्दी से आता है। ये बहुत ही अच्छा एंटी इन्फ्लेमेटरी प्लांट में लोग क्या करते हैं, महुआ की शराब में उसको डाल के पीते हैं, तो जितने भी शरीर के दर्द है, जितने भी शरीर की सूजन है, वो सब ठीक हो जाता है। तो बहुत जमाने से पहले से दारू हल्दी यूज होकर आ रही है, लेकिन इसको दारू हरिद्रा, इसका संस्कृत नाम है दारू हरिद्रा। दारू का अर्थ होता है वो गुड़ से कहते हैं दारू, दारू का मतलब होता है गुड़, यानी जिसकी लकड़ी पीली हो हल्दी जैसी, वो दारू हल्दी, दारू हरिद्रा। उसकी लकड़ी होती है बिल्कुल पीली हल्दी जैसी होती है। जी डिकॉक्शन बनाते हैं, शराब में खूब यूज करते हैं, हमारे भगवान में खूब पीते हैं लोग।
हॉप्स के जो फ्लावर होते हैं वही है?
जी सर,
हॉप्स?
हाँ हॉप्स नहीं है सर,
अच्छा,
दिस इज नॉट हॉप्स, दिस इज ये तो वो है क्या बोलते हैं बारबेरिस, जो हमारे केसी मेहता साहब का जो प्लांट था बारबेरिस, पचमढ़ी में उन्होंने बहुत काम किया था उस जमाने में। सॉरी जी सर, हरड़ हरड़ हरड़, हाँ हरड़ा बीजा और मेरा छाल, ये भी बहुत इंपॉर्टेंट प्लांट है। फिर यहाँ पर कुछ ऐसे प्लांट्स हैं इसके बारे में शायद आपने कभी कल्पना ली होगी, 5 मिनट ओके सर ठीक। ये प्लांट मैं आपको बता रहा हूँ, इंसेक्टिवोरस प्लांट भी आपको सुना हो कि ऐसे प्लांट जो स्टिंगिंग सेल्स होते हैं, आप छूते हैं आपको डंक मारते हैं, तो डंक मारने वाले पौधे जो आपको मिलेंगे, हिमालय मिलते हैं, पछाड़ी मिलते हैं, आपको फटाफट दिखा देता हूँ जानवर हमारे यहाँ क्या क्या मिलते हैं। 5 मिनट के विदिन 1 मिनट में आपको बात करता हूँ।
Thank you, sir. There are nice presentation about biodiversity of Panchmani by Dr. Mutations, sir. Thanks.
8 मिनट लग गया, कन्वर्ट एक्स्ट्रा लग गया, सॉरी। धन्यवाद आप सबको सर, मुझे सुनने में बहुत अच्छा लगा सर।
Thank you very much, sir. Now we have Professor Dr. I.S.G.B.R.D ma'am from Principal Scientist from BRMR Bharatpur Sevar. Ma'am will be presenting her paper on morphophysiological and metabolic response in rapeseed mustard after irradiation with mutagenic agents. And you want to ma'am.
Good afternoon to you all, and I welcome chairperson. So respected chairperson, co-chairperson and dignitaries present in the hall, I am going to present the work regarding the morphological, morphophysiological and metabolic responses in rapeseed mustard. So first of all, what I will do, I will give you some insights regarding the mustard production, and then I will go for the biochemical analysis, which I have conducted. So rapeseed mustard ranks second and third in the Asian production respectively. And it is very important because it offers high return with a low cost of production. This is the production trend in world. This is the production trend in India. This is the production states, different states of India. Total oilseed production, and this is the edible oil availability, share of rapeseed mustard, oil import and consumption. And this is oil meal. This is very important because here we are working on the oil meal export. So we have to enhance or increase the oil meal export by removing the anti-nutritional components present in the meat, because meat has 2 very important anti-nutritional factors. These are the major rapeseed mustard producing countries, and this is the demand, availability and per capita consumption of edible oils. This is the area production yield in different scenario, like in 2020, 30, 40 till 50. We have already made the roadmap for 2050 for the production and productivity of rapeseed mustard in India. And this is the total share of vegetable oils in total imports during 14-15. This is again the export import of refined or rapeseed mustard oil. And these are the major states in India which are contributing in RM area and production in India. And this is very important because it shows that RM is having antioxidant activity as well as it is having an inhibiting cancer cell proliferation activity, which is very important. Now the fatty acid composition of RM is what? That is the mustard oil. मस्टर्ड ऑयल सरसों का तेल जो आप लोग सब घर में खाते हैं, उसमें हमारे को क्या क्या मिलता है, फैटी एसिड हम क्या क्या खा रहे हैं, दैट इज वेरी इंपॉर्टेंट because as the previous speakers they told us regarding the immune system. So this fatty acid, or you can say mustard oil जो आप लोग घर में खाते हैं, that is going to impart the or boost up the immunity, human immunity. So in this case you will find that it is the erucic acid. If you look at these different fatty acid, palmitic, stearic. These are the major fatty acids, and out of this, erucic is having 40 to 55% in the range, and which is highly toxic. So mustard oil, सरसों का तेल खाना बहुत अच्छा होता है, ये हम लोग बचपन शुरू से खाते आए हैं. But the major main important point is that erucic acid, if it is present in high amount, it will be toxic to the body. So crucifer as a source of bioactive compound, as I told you that these are the three major components which are very toxic, which are present in the mustard oil. These are the glucosinolates, fatty acid like erucic acid, only erucic acid, not other oil fatty acid. And apart from this, the phytic acid. These three are the anti-nutritional factors which are present in the mustard oil. Otherwise, mustard oil is having anti-cancerous property which is not present in any of the other edible oils. Apart from this, the omega-6 and omega-3 ratio, that is mu/pu ratio, is only present in the balanced amount in case of mustard oil, not in the even olive oil or जो भी आप धारा या जो भी लेते हैं उनमें किसी में भी present नहीं होता है. And it can be also used as a probiotic. Now metabolites which are major metabolites which are present in the mustard, that is the sinigrin. Sinigrin is very important, as I told you that sinigrin is responsible for the pungency of the mustard oil. तो जो सरसों का तेल को हम खाते हैं पकाते हैं तो उसमें एक घास आती है, कहते हैं घर में उसकी स्मेल भर गई, वो जो घास है वो sinigrin से है. Sinigrin is very important क्योंकि अगर वो घास नहीं होगी तो sinigrin कम होगा, और वो sinigrin जो है वो एंटी कैंसरस सब्सटेंस है. ठीक है, तो घास इज वेरी इंपॉर्टेंट मस्टर्ड की. तो अब जैसे हम कई होटल्स वगैरह में जाते हैं, हम कहते हैं कि मस्टर्ड ऑयल से बना हुआ खाना है, लेकिन उसमें वो स्मेल नहीं आती है. एक्चुअली उन्होंने रिफाइन कर दिया उसको, रिफाइन नहीं, सिनेग्रिन को उन्होंने खत्म कर दिया. सिनेग्रिन खत्म हो गया मतलब उसके अंदर जो घास है वो तो कम हो गई, लेकिन उसके न्यूट्रिशनल कैरेक्टरिस्टिक भी कम हो गया. So this is the two major anti-nutritional components. These are the sulfur-containing compounds, that is glucosinolates, and apart from other, that is the phytic acid. And so three major anti-nutritional factors are there: erucic acid, glucosinolate, and the phytic acid. So our main motive is to lower down the erucic acid, to lower down the glucosinolate, and to lower down the phytic acid from the mustard oil. So what will happen कि जो भी मस्टर्ड ऑयल उसके बाद आपको मिलेगा वो पूरी तरह से प्योर होगा, पूरी तरह से उसके अंदर घास भी होगी, प्लस उसके अंदर एंटी न्यूट्रिशनल कंपोनेंट्स भी होंगे, और घास भी उसमें कम होगी. These are the major role in human biology, that is again the human biology pathway. These are the anti nutritional effects, and so considering all this thing, what I did, I took the two major genotypes from the PM21 and PM30. These are the low erucic acid and low glucosinolate content, but phytic acid was more. So what I did, I went for the irradiation with the gamma irradiation, 1000 gray and the 1500 gray, as well as the chemical treatment, that is the EMS, ethyl methyl sulfate treatment. And then these are the different experiments which I designed, and then I studied the germination percentage, viability of the seeds and the aborted seeds. And then these are the figures where I found the mutation in the way like sarson का फूल आपको पीला दिखेगा, but after mutation I also received white flower plants. इसमें एक plant है, I think it is visible from there also. White flower is also there. Apart from this, so many variations are there, and these are the mutative mutants which are identified in M4 generation. M5 generation we have already grown now, and after the biochemical analysis, then we will confirm the results. And these are the PM30 results also. And these are the characteristics which we have studied, that is the dry matter, crude protein, ether extract, nitrogen-free extract, and so many biochemical parameters we have studied. Now another point is that this is the case of the genotypes which are going to produce this oil, mustard oil, sarson ka tel. But what about the khal? Khal, you know that it is a machine. Khal is required for the animals as an animal feed. अगर उसमें घास ज़्यादा होगी तो एनिमल उसको नहीं खाएगा, गाय उसको नहीं खाएगी, भैंस उसको नहीं खाएगा. तो उस केस में हमें ऐसा कुछ करना है जिससे कि हमारा जो मस्टर्ड मील है उसके अंदर वो एंटी न्यूट्रिशनल कंपोनेंट्स खत्म हो जाए। तो उसके लिए हमने इरेडिएशन दिया। इरेडिएशन देने के बाद हमने उसके डिफरेंट मेथड से कंपाउंड्स को सेपरेट आउट किया, एक्सट्रैक्ट आउट किया, उनको स्टडी किया कौन कौन से ग्लाइकोसाइड्स हैं, थायरोल्स हैं, स्टेरोल्स हैं, और उनकी न्यूट्रिशनल एनालिसिस करी कि ये टोटल एंटीऑक्सीडेंट एक्टिविटी एंड सो ऑन. And similarly, we also carried out the phase contrast microscopic image of the seeds which were irradiated. So it was found that flaring was there after the irradiation. That is also one of the very important aspects. And when we went for the genetic study, also this work is is going on these days. So in this we went for the QPS, for the aliphatic glucosinolates. And the molecular study shows that 12 GDR orthologs we have, out of the 12 GDR orthologs, only one is mutated after the irradiation with the 1000 ray, 1500 ray, as well as the EMS treatment. And we were able to reduce approximately 60 to 70% of the glucosinolate level along with the phytic acid in one of the two genotypes in Brassica. That is very important thing because, but we cannot go for the publication or we cannot go for the registration because still as cell generation, if it is consistent, then we will go for the registration and the publication. So these are the major deliverables where the variety low glucosinolate, low phytic acid, and low erucic acid will be developed. Although low erucic acid and low phytic glucosinolate varieties are already there, you must have heard about the canola varieties. Canola varieties where glucosinolate is very low, that is less than 30 micromole per gram, as well as erucic acid is very less, and that is less than 2%. But now we have reduced down the phytic acid also. So these are the future prospects where cytoprotective effect of the hydrocinamic acids will be studied, and it has been seen that if we consume the mustard oil which are having low erucic acid, low glucosinolate and low phytic acid, then it is going to be boost up the immunity of the body also, human body also. And this is the acknowledgement because for this work I got financial aid from the VRNS, Department of Atomic Energy, and the I.C.B.R.D, that is Indian Council of Agricultural Research, and Directorate of Deep Sea Mustard Research in India.
Thank you, madam. Very nice presentation and introduction of this mustard research for your contribution. As a human being, I think in future we get some more varieties and some other knowledge. As a accomplished background student and scholar here, so lot of thanks to you for your nice work and congratulations for this nice presentation. Thank you very much. The next speaker is Dr. Pankaj Gupta ji from USA.
Hello everyone, can you hear me?
Sir, just give me a minute. I'm just
Sure.
We have Dr. Pankaj Gupta, Principal Scientist, Biotherapeutics Discovery from USA. Sir will be presenting his paper on engineering approaches for development of biotherapeutics. Sir, we have with us the chairperson for this session, Professor B.K. Chaturvedi sir. We have the co-chairperson, Professor D.S. Chokar sir, and the reporter, Professor Manoj Pandey sir. They will be chairing the session. Handing over to you. Thank you.
Thank you everyone. Hello, can you hear me?
Yes, sir. Yes, sir. Yes, sir.
Wonderful. Good morning, good afternoon, good morning here in US. I don't know if anyone is here from US, but good afternoon to friends back in India. And today I'm going to talk about engineering approaches for development of biotherapeutics. Before I begin, I'd like to thank the host for inviting me and my
Sir, please make your screen, your presentation full screen, sir.
yeah,
Full screen.
actually I have multiple screens and it will go in a different screen. Let me see
Okay, okay, okay.
if you see this. Now, do you see
Yeah,
this?
yeah, yeah. It's fine now. It's fine. It's
It's
okay.
fine with you?
Yeah, yeah, yes, sir.
Okay, wonderful. So today, as I mentioned, I'd like to thank the host and I will talk about engineering approaches for development of biotherapeutics. And before I begin, I work with this pharmaceutical company called Boehringer Ingelheim. And I'd like to briefly tell about Boehringer Ingelheim. It's a family owned global corporation. And this company was started in Ingelheim in Germany in 1885. And we have about 50,000 employees worldwide, and with an annual sale of about 20 billion euros. And within Boehringer Ingelheim, we are like a small biotech company called Biotherapeutics Discovery, and this Biotherapeutics Discovery is a global function based in Ridgefield, Connecticut, USA. And here is a bird's eye view of our building. We are located somewhere over here, in this particular complex. And we have three major research sites. One is in Vienna, second one is in Biberach, and third, of course, in Ridgefield in USA. And Biotherapeutics Discovery supports all of these research sites. We work very closely with them, and as I mentioned, that Biotherapeutics Discovery is like a biotech company within a pharmaceutical company. And this department was initiated in 2007, and initially the name of the department was Global Skill Center. And we started working on making antibodies for human usage in 2007. And our first success came when we had this molecule called BCMA-CD3 BITE. So this is a bispecific SCFV based antibody that we created, and it went into clinic in around 2015. And in the same year, we got our first FDA approval for the drug called Praxbind. So Praxbind is basically it acts as an antidote for our molecule called Predexa. Predexa is basically a blood thinner, and there were some incidences where patients saw heavy bleeding. So to avoid that heavy bleeding, our department created Praxbind, and this came as first success in 2015 when we got U.S. Food and Drug Administration approval. Our major success came in around 2016 when this antibody called risankizumab first went in the phase 3 trial. So for people who are not aware, with the drug development, we are first in the research stage and then in the development stage, and then we enter into the clinical stage. And in the clinical stage there are three stages: phase 1, phase 2, and phase 3. And once we see efficacy in phase 3, we get approvals from FDA. So our first antibody, major full antibody, went in phase 3 trials in around 2016, and then in 2019 we got our second FDA approval for risankizumab, and currently this antibody is marketed by the name Skyrizi. And around the same time we were working on a third antibody, which was against IL-36R receptors, and the name of this antibody is, internal name is BI655130. And then we got some very good results, and now I want to show you over here, as picture is worth thousand words, that we went in clinical trials with this antibody IL-36R for a disease called psoriasis. And as you can see that we gave a dosage of 40 milligrams to a patient, which is patient 2 over here. And if you see, from day 1, which is a baseline, to day 28, which is week 4, you can clearly see the efficacy of our antibody IL-36R, antibody spesolimab. And I'm very happy to share that recently we filed for a BLA application with Food and Drug Administration. Basically, BLA application is the last stage where a drug company can do for approval of a drug. So recently we have filed for BLA in 2021, and we are looking for approval of this drug soon. So having said that, what I want to emphasize over here, or what I want to inspire young budding scientists in this college, is that biotherapeutics is something where we use our recombinant DNA engineering to create a drug candidate. And for making a drug for human consumption, either we can have a drug in human body, which will be unethical, or we can go ahead with some species. So what we do is we go with immunizations in mice, rabbit, llamas, chicken, and we try to use all antibody repertoires to get the drug candidate, or to find that candidate which can have drug-like properties. So what we do is, once we have immunized the animals, we take out the spleen, and then we remove the B cells, and then we evaluate the B cells by flow cytometry, and then we try to look for binders for our antigen. And once we have evaluated the binders, we sort those cells, and then once we have sorted those cells, we isolate the DNA from them, and then recover the antibody sequence, which we call the variable region recovery. We recover the V regions from them, and then we try to evaluate these molecules. Having said that, the molecule is still not for human consumption, and molecule has to be made a drug. So right now, even if we get a binder, it is just a binder. We really need to identify how can we make a drug out of it. Apart from using the animal based drug generation, we also used um, a technique called phage display. Phage display is a very in vitro based technology where we can have a phage expressing everything on the surface of the E. coli, and then these particular E. coli can be screened to identify a binder. And we use this library called Superhuman Library 2.0 from a company called Distributed Bio based in California. And once we have identified the binders, again, these binders are not meant for human usage. So how do we proceed ahead? So our strategies are different now. So 10 years back, the strategies which the industry followed was that we will we will immunize animal, create hybridomas, screen about 100,000 hybridomas to identify about 1,000 binders, and then go for a functional antibody characterization and identify about 1 to 5 candidates which will have drug-like properties. But today, the focus has been shifted, and now the important parameter for us is not only to have a good drug, but also to have a good drug like properties. So that's one of the most important feature which I'll be talking about in today's talk. That great, we have identified a wonderful binder, but what do we do with this binder if it goes in pharmacy, and just before it is given to a patient, it clouds, it becomes like a milk, or it becomes like a curd. So then it's of no use, the molecule will be thrown away, and it is not fit for human usage. So uh, to our rescue comes the biopharmaceutical informatics. What does it tell us? That we can, at the very early stage, when we are identifying the drug candidates, we can go for evaluation of those candidates on basis of multiple parameters, and we can take into account CMC properties. CMC is chemical manufacturing and controls on these candidates. If we can really look into the CMC properties of the potential drug candidates very early in the game, then it can really solve a lot of problems and lot of expenses which a pharmaceutical industry has to incur to make a drug candidate, and eventually it will solve the pricing for the drugs. So now I will talk uh, about how we make a drug from a tool that is a binder and that has some properties of becoming a drug. My group focuses on lead optimization, and lead optimization is a process of converting non-human sequences to be more human-like. while maintaining the key attributes such as binding and function. And our philosophy is that we want to retain the binding as like a parent tool candidate, and we want to have a minimal change in function. We want to have lowest potential for immunogenicity. And by immunogenicity I mean that when the drug is injected in human body, we don't want any untoward experiences in human body. And to that effect, we use a software called Apivex, and this Apivex can really identify T cell epitopes in the particular um, sequences. And of course, as I mentioned earlier, that in very early in the game, we look at the CMC properties of the molecule, so that there are no predicted glycosylation sites or deamidation. Deamidation is when the amine is removed from the protein amino acids, and as a result of deamidation, what it can do is it can cause a heterogeneity in the protein, or also it can create isomerization. As a result, we will see lot of precipitation in the proteins. And we have recently identified that it's generally, these are there are 2 to 6 amino acids which are critical in the mouse residue or mouse sequences, or let's say llama sequences, which are essential to maintain the binding, and remaining other residues in the amino acid sequence can be converted to a human like residues. And for that we use combinatorial approach. What we do is we try to put a choice between a human and a non-human residue, and then try to screen that which residue will be important for binding. And how we proceed is, and what all features do we take into account during this lead optimization, is that we really want to remove some of the challenging regions from the sequence. We want to keep developability intact for the molecule so that it can have good drug like properties. And of course we don't want to lose the binding and function of the tool candidate. And to do humanization, what do we look for? We look for any sequence which is more than 85% similar to the human antibody sequence or human germline sequence. To that effect, when we say that we want to make it more human like, we do CDR germlining, and we have seen that sometimes not all residues in the particular sequence are important, and we can change it to the human sequences. So this process is called CDR germlining. So we do CDR germlining. And then also what is important is that we want to keep the immunogenicity as low as possible. And as I mentioned earlier, we use this tool called Apivex, where we want to keep the scoring below -20, so that our human body doesn't recognize it and doesn't perceive it as foreign. And most of know that antibody is a Y-shaped molecule consisting of 4 different chains, 2 light chains and 2 heavy chains. And once whenever we run an immunization campaign in a mouse or non-human, and the reason why I'm saying mouse is most of the time 95% of the time we use mouse because of the ease of handling. But let's say if we see that mouse cannot be used, then we go for chicken, or we go for llamas or any other species. But let's say, having said that, let's say we get a tool candidate which looks like which meets the binding properties of the molecule. Then what we do is we identify the variable regions, and these are the variable regions. And what we do is we transfer the variable regions to a human antibody, where we have constant regions from human. And these constant regions from human, once this antibody is created, this becomes a chimeric antibody. Now this is a mouse human chimera, and this antibody is evaluated for all the binding properties and the functionalities, and if it meets the functional requirement of the antibody, then what do we do is then we take the variable regions of the antibody. And then now I'm trying to convert, what I can show over here is that we are trying to convert all the red kind of sequences from a mouse to more human like. And as you see that gradually I'm transitioning to to bluish areas, so that we finally come up with the sequence which is completely acceptable for human body, and it still retains the functionality and the binding properties of the original tool candidate. And to that we use a lot of techniques, but most of the all we use phage display. Phage display comes in very handy for us, where phage display can be used for different methodologies. We can go for soluble screenings of the E. coli, or we can go for a complete display. So if we want to go for a soluble screenings, what we do is we create, let's say, this is a mouse sequence, and in the mouse sequence, what we do is we create point mutations to make it more human-like. And the way we do is we use overlapping oligos, and then by PCR-based technologies, we can bring all the mutations together, and then we clone them in a M13 phage based vectors, and then this phage DNA is transfected, E. coli is transformed with this, and then there are two possibilities. Now, either the phage can display the protein onto the surface of E. coli, or it is also secreted out in the media. So what we do is, at this stage, if we have a small diversity, if we really want to look at couple of residues that we want to change, and our diversity is like 5 into 10 is power 3, then we can quickly go for phage mid screening, phage screening. And in that, what we do is we grow E. coli cultures, and then take the supernatants, and then these supernatants are evaluated by ELISAs. And we can do this kind of lead optimization in about 3 to 4 months. And then and of course, our we use robotics, to really go for um, high throughput experimentations, where we put the plates, and then program the robots to identify the plaques, and then these plaques are put in separate wells, and then they are evaluated by ELISAs. And so this technology works very well if we have a small diversity. We can really construct a library, and we can evaluate them in ELISAs. However, let's say, if we have to really, the species is very far away. For instance, if we are going for camel, or we are going for chicken, and now really we are very very far away from human, so we really need to make a very big library to the range of 10 raised power 8. And what happens is that, if we have such a big library, then phage mid screening will not work. Then we use the power of phage display. And phage display is such an extensive technique, where a binder can be expressed on the surface of E. coli, and then if we do kind of panning, we can really identify the binders. And phage display enables us to screen a diversity of about 10 raised power 8, although people have even tried higher. But in my group, we have tried up to a diversity of 10 raised power 8. And then, as a result of phage display, initially, in the first rounds of panning, we will have a big diversity. But then, as we move towards the round 2 or round 3 of panning, we can really have a targeted uh, identification of the molecule, which is a high affinity binder. And the way we do engineering, and our criteria is that, we initially try to put the binder on a human framework. We try to identify the germ lines, human germ lines, that can really retain the binding, and we can put the CDRs on the human germ lines. And in the next stage, we try to remove all the potential challenges in the sequences. For instance, asparagine and threonine together makes a deamidation site. So for instance, in this case, uh, asparagine 52 was identified to be a challenge, or methionine 58 was identified to be a challenge, or likewise, aspartic acid and glycine together forms a deadly combination at in the H.G.B.R.3. So what we try to do is we try to remove these potential challenges, and then finally we triage the hits on the basis of binding analyzers. We look at the percent humanness, and as I mentioned earlier, that we want our drug candidate to be more human like. So we really go for identifying anything which is more than 85% human. We look at isoelectric point of the variable regions, and once we have identified certain handful of candidates, we try to format them as IgGs. We identify about 30 to 50 candidates, and then these candidates are put to test. We really evaluate them on the monomer content, so how they come out of the purification column. That's very important, because let's say this is the first criteria when a molecule is put on a purification column, and if let's say the percent monomer content of the molecule is low. For instance, in this case, you see there is a candidate which is 36% monomer as it is coming out from the protein A column. So this is like a bad indication that this molecule is not going to be stable. We aim for anything which is more than 95%, and over here you can see that we look at the percent monomer content, then we look at the stability of the molecules, and we try to aspire for anything more than 60 to really the TM of the candidate. We look at the hydrophobicity of the candidate, and then on the basis of all these parameters, we try to look for candidate by our bioinformatics pre-formulation assessment, which is green all through. Because if anything, because this is the stage when we are doing in the drug discovery space, we can really look for candidate which has good properties. So so right now I talked about the tools that we use in our processes. Now let me talk about really creating a drug candidate. How do we create a drug candidate and the basic biology behind it? So now I will talk about engineering approaches for development of novel T cell engagers. And as most of the presenters over here, they have showed that immune system is of our body is very very critical for our well-being. So what does immune system do? Immune system basically it recognizes anything which is foreign, and it kills them and it removes them. And also immune system does it recognizes anything foreign from self, so it has a power to recognize between self and non-self. And how does it do is, we can harness this power of immune system to really create therapeutics for our human body. And how does that work? For instance, now since T cell engagers are very well used in cancer immunology, I will talk about how we can develop therapeutics for cancer therapies. So now there are traditional cancer therapies like chemotherapy or some other therapies, where what we do is we try to target tumor, and we give chemotherapies non-specifically to really kill the cancer cells. However, if we can program our immune system so that immune system becomes the drug, then this will be really helpful for our body. This can really target the host, and it can really target the same areas in the body which is really where the drug is needed. So this is the part of the cancer immunology that we are currently supporting. And just to give a brief description, cancers can be of multiple type. They can be cold cancers or hot cancers. And by cold and hot, I don't want to go and with the literal meaning, but what we want to tell over here is that if the tumor is immunoreactive, then it can be really programmed, and it can really kill, uh, reduce the tumor burden by the T cells. But what happens during cold tumors? Cold tumors generally happens then when there is a deficiencies in T cell priming, and tumors create a very immunosuppressive environment, and this immunosuppressive environment really doesn't allow any immune system to come in the tumor vicinity to really kill the tumor. But immunotherapy has a potential whereby we can
Hello.
we can turn cold tumor into a hot tumor.
Hello.
Yeah,
Dr. Gupta, yeah.
can you
Sir,
hear me?
please conclude in 10 minutes. It was
Okay.
little
Yeah, no
late.
worries. I'll be done.
Okay.
Yeah. So what we are saying is that, if we can create bispecific antibodies that we can bring them in the tumor environment, then we can really shrink the tumors. And over here, as I said earlier, that, picture is worth 1,000 words. Over here we are showing the tumor cells, and then we are trying to bring T cell in tumor vicinity. And our first generation of T cell engagers that we developed were BCMA BITE in 2015. However, these first generation tumor T cell engagers had a lot of challenges. There was a PK issue of this molecule, and then there were CMC issues with the molecule. So we came up with a new format, our next generation format, where we could target the tumor, and we can have a T cell engager on the other end of the antibody. And how we did was we created this molecule which we called as a doppelmab. Doppelmab in German means bispecific. So we went ahead with this molecule, and our whole rationale was that, we wanted to increase tumor localization so that we can have multiple target binding arms, and we can have a T cell engager on the bottom. However, if we put two T cell engagers, then, it can be really toxic to the patient. So we really wanted to play with the T cell engager part, so that, we can reduce the affinity of this molecule without compromising the efficacy. To that effect, we engineered linkers. First of all, we tried engineering linker. We used these glycine serine linkers. So we tried different length linkers. And in the interest of time, I will not go into the details, but all these molecules were expressed and purified, and we got very good purification yields. And then we next moved and we evaluated these molecules for their stability. And as you can see over here, the stability of these molecules was really low. We could, with the 20 length linker, we could reach an affinity value of about 53 over here. But we really wanted to achieve anything which is higher than 60 or something around 60. So then what we did was we created a disulfide linkage between the variable region residue 44 and the kappa residue 100. And this disulfide linkage was we purified the molecules again. And as you can see that with the addition of the disulfide linkage, we were able to improve the TM of the molecule from 54 to 59. And for a drug candidate, this improvement is quite large, although it might seem trivial by looking at just by that, we are able to improve by 6 degrees. But for a drug candidate, it means a make or break case. And then we evaluated the binding efficiencies of all these linkers. And as we found that, the binding was really improved by addition of the longer linkers. And and we went ahead and we evaluated the molecules by flow cytometry. And as you can see over here, we over here now I'm showing a wild type doppelmab over here in the first column, and in the second column I'm showing a doppelmab where we incorporated a disulfide linkage. And as you can see that, with the incorporation of disulfide linkage, we reduced our binding affinity from 1 nanomolar to something like 127 nanomolar. So this was something we were looking for. We really wanted to create a molecule with the low binding affinity. Otherwise it could have been a toxic molecule for human body. And then we evaluated these molecules in our functional assays. And over here, please pay attention to the green and the blue curves compared to purple and the black curve. So black curves and the purple are the doppelmabs, and then the ones with the green and blue are with the disulfide linkage. And as you can see that, the molecule that we created by putting a disulfide linkage had a higher potency compared to the parental molecule. And although these molecules, they were lower affinity, but they showed better potency in our functional assays. And then we evaluated the non-specific killing of all our molecules, and we did not observe any non-specific killing in these molecules. So this was the good thing. And now I will like to summarize that, I have shown you our robust process for identifying binders that matches industry standards. We try to take into account valency, affinity, and developability in designing our drug candidates. We over here I have shown you a case study where we improved the stability and CMC property of a CD3 binder during our allo process. And also these anti-CD3 binders depict better properties in terms of no non-specific killing. And I would like to end my talk with one last statement that, the choices what we make in our drug discovery about molecular design and engineering greatly impacts the path to clinic. And we are currently working with our therapeutic areas to move these doppelmab molecules into the clinic to see the impact in patients. So I will end here. And these are some of the members of our team who have worked diligently to create these molecules. And I'm ready for questions.
Yeah.
Thank you.
Thank you, Dr. Pandagupta, for your work in field of this molecular engineering and biomedical therapeutic. And I think all the audience and here in this hall related to topic get benefit from your talk. I think if any query and question from the audience, they can make question to Dr. Pandagupta.
So is there any
So
questions?
I think there is no question. Thank you very much for your valuable time here. Actually, we are very late in the evening here, and some more speaker also in queue. So in last again, many thanks to you, Dr. Gupta.
Oh, no worries.
Oh,
My
no worries.
pleasure.
My pleasure.
Thank you very much, sir. Now, we have our very honorable lead speaker, sir, from IIT Roorkee, Professor G.S. Randhawa, sir. Before starting his presentation, I would like to felicitate a few of our technical session chairperson, co-chairperson, and reporters. There were 2 technical sessions running parallelly, and also a poster presentation session was also being run in the next hall and the next to next hall, this the media center. And for that, I would request we are waiting for the chairperson of that parallel session. Soon she'll be joining us. And for that, I would request Professor Shurveer Singh sir and Dr. P.K. Upadhyay sir to please felicitate our chairpersons which are going to come. Sir, you may please call the chairperson of the poster presentation, Dr. Anbhuti Sharma. Sir, just 2 minutes. Sir, just 2 minutes. I would like to invite with the co-chairperson of the technical session which was running in the next hall, Dr. S.P. Singh sir, Principal Scientist from I.S.G.B.R.D. Gwalior. I would request Honorable Arun Chopra sir to please felicitate Dr. S.P. Singh sir with the Patka cloth and token of gratitude. Sir has co-chaired the session, the technical session 4, which was running in the next hall. A huge round of applause for that. Also, now I would request, we have with us the chairperson for the poster presentation session. I would request Professor Shurveer Singh sir and Dr. Arun Chopra sir to please felicitate the chairperson of the poster presentation session, Dr. Anubhuti Sharma ma'am, Principal Scientist, DRMR, Sevar Bharatpur. A huge round of applause, please. Dr. Anubhuti Sharma ma'am. In the sequence, I would like to request the co-chairperson for the session, Dr. M.D.-Bharti sir, to please come forward for felicitation. Dr. M.D.-Bharti sir, District Fluorescent Officer from MP Dhar. I would request Professor Shurveer Singh sir and Dr. Chopra sir to please felicitate Dr. M.D.-Bharti sir with the token of gratitude and Patka cloth. And next in the sequence, I would like to invite Dr. Pramod Kumar Prajapati sir for the felicitation. He was the reporter of the poster presentation session. A huge round of applause for sir as well. Great recommendations were given for the poster presentation, which will be discussed in the valedictory session. Thank you. Now we have our next guest speaker, Honorable Professor G.S. Randhawa sir from IIT Roorkee. Sir will be presenting his presentation on recent developments in molecular mechanism of protein transport in a living cell.
Good evening, everybody. Good evening, everybody. I'm thankful to the organizers for giving me opportunity to share my views in this very important conference. I'm especially thankful to Dr. Arun Chopra ji. This topic which I have chosen just a few years ago, I thought that something new in plants I would start, and so this I thought was my first lecture at Goa in when I made. I was just expecting so many of our publications were in pipeline, and then somehow the things changed a lot. Then this COVID problem came. I retired in 2018. Lost touch with my students, so couldn't make breakthrough. But still the topic remains very interesting, because here I found that there are many presentations on plants. So all those who are working on plants, for them this is a new idea, some new field, because there are very few publications in this case. Particularly our students, I am addressing to all the students here, faculty members, they are very familiar with protein synthesis, translation. The protein is synthesized, and then they are familiar with some signal sequences. With the protein, there are some signal sequences, and that's all. Something very important which didn't come to attention of scientists, or rather very late this research in other organisms started, that when protein is synthesized in a cell, when translation takes place, so protein is not required at that site. So how this protein moves to another site in a cell. So I'm not talking anything, protein traveling in vessels from one place to another, or moving from one set of cells to another set of cells, or something like that. So all that what I'm talking is inside a cell. So all this talk is inside a cell. So protein inside the cell is produced at one point, and then how it is sent to another site within the cell. So when people move their attention to this and slowly they start putting together these pieces. Can you get water? Welcome.
सर इधर रखा हुआ है
When protein is transported from one site to another, there are many mechanisms. So here I am talking about just specific one of those. So this mechanism is just like we are transporting grains or some other material like that transportation. So here I am talking about vesicular trafficking, vesicular trafficking. So that protein which is to be transported, it is enclosed in a vesicle. Then from one organelle, it is sent to another organelle. You can imagine there are thousands of other proteins. So this protein has to be picked up and then it is to be taken another side. Then it shouldn't be damaged at all. And there no set path or something, it has to be carried through a certain path. And then it has to be delivered at a particular point. If there is a mistake at any of those points, the whole machinery goes wrong. There are problems, serious problems with the organism. So what thought I am giving to the breeders, to all those who are working with plants, so that if they are interested to bring further improvement, then they should look at all these transport genes. So there are several proteins which take part in that, and there are several genes coding for those proteins, and people looked at the sequences, and we also looked at many sequences, many regions of that. So there are certain sequences which are very important. There are certain sequences which are very vulnerable. They can mutate. So when the breeders have their material, they should see, and then they should select that material based on that it should have healthy genes, transport gene for this protein transport. So it is such a vast field that I can talk about several hours on this, and it's so interesting, very, very interesting that how this is going on. Just some fundamentals I would touch here, and so transport of a protein from one location to another is required for the normal functioning of a living cell. The transport is achieved by several mechanisms. As I told you that vesicular trafficking is one of the mechanisms. What is vesicular trafficking? In vesicular trafficking, the protein is enclosed in a membranous vesicle and transported from one compartment organelle to another in a cell. So this vesicle is interacting with a specific acceptor compartment. So when this vesicle is moving from first place, first it is formed, then it is moved, transported when it reaches. So there is a specific acceptor compartment by site-specific signals. There are certain signals. By the help of those signals, these vesicles are assembled. So how that protein is selected, again with the help of a certain signal or certain set of signals, so it is selected. And after that vesicle formation, it's all with certain sequences, and then it moves further, and at the site of delivery, again there are very crucial signals, and then it is fusing with the membrane and then delivering the protein. Vesicular trafficking takes place along the exocytic, secretory and endocytic pathways. So let me inform you that 2013 Medicine Physiology Nobel Prize was given in this area. So just few years back, Nobel Prize was awarded in this vesicular trafficking region for physiology of medicine field. So here just a brief view of that you can have. So you
can use that this one mouse. Yes.
So it's very simple. You can see that there is a starting point So on that side, the protein, and you can see there is a soluble material which is to be transported, and then there are membranes embedded in the cytoplasmic membrane organelle membrane, and when these proteins are vesicle is to be formed, so there are several players, several components there. So I have shown some of those components. From those components, okay. Yeah. Okay.
आ रहा है हल्का है ना इसलिए देखने लगे
So anyway, this is not much need I can explain. So here you can see membrane proximal coat protein, membrane distal coat protein. So when that vesicle is formed, so it has the inner layer, then there is the outer layer, very interesting subject, very beautiful structures, these are formed, and then there are other small GTPAs. So how these are organized, they recognize each other. So the inner layer is formed, so slowly budding takes place, then the bud enlarges in size, then this vesicle is formed from these components, these proteins, and then there is a detachment of that vesicle, and then you can see that there is an uncoating taking place. So then the movement, it goes cytoskeleton, along that this movement takes place, and finally when it reaches the destination, so at the destination again there is a recognition of a particular signal and how it is delivered. So there is a particular, you can see that protein, how the other proteins from this side, they are recognizing each other. So this is like security people are checking that when you start from the starting point, so there is a tag, so that tag is recognized. From recognizing, it is picked up, and then the vesicle is packed just like at airport. You can imagine the scene at the airport. So then it is carried forward, and then again those coats are removed, and then finally it is delivered. So this is very complex and so many proteins and so much, and accurately this has to be done. It's just diametrical.
Sir, is it exactly same mechanism by which the process of endocytosis occurs, or is it different than that? Because there also all these steps occur.
Yeah. I can't compare, but here all the players I'll go no further to molecular details, those chemicals which are participating here. One by one they were discovered. So this discovery started around 1970. This first one, the protein was started around 1970, and the other major player we call adapter protein complexes. So they were discovered later on around 1990 or somewhere toward the end of the last century. One by one, they were discovered. Let's see the complexes involved in trafficking of protein. There are coat proteins, namely COP1, COP2, so one category. Then there are adapter protein complexes, we call AP complexes. For the youngsters, many of these names would be new, and for all plant scientists, these things are new. and there's a lot which could be done on that. And the third important protein, that is the accessory protein. Accessory protein. So the most famous accessory protein is clathrin. So very first discovery was in England, UK, and they started from the slaughterhouses, from certain animal parts, from sperm and other things. One by one, these things were discovered. So three main categories of the proteins, COP1, COP2. Then adapter protein complexes. Third is the clathrin and other accessory protein. Clathrin is the most famous. The study or the history of each of these molecules is very important, and I had a lot of interest, or some of we couldn't make that here. So COP1 is code protein, mediates transport of a protein from Golgi to endoplasmic reticulum, and COP2 code protein mediates transport of a protein from endoplasmic reticulum to Golgi. So So here, just to show the COP1, COP2, this is between endoplasmic reticulum and Golgi, just showing that role of those proteins. Very interesting thing, you can imagine yourself also, those pieces are lying there. So these sacs are not that sacs are first made ready, it is brought to the site and then something is packed into it. No, it's not like that. So this is the protein to be transported. There is a specific sequence. On this the protein will identify. So it will attach. So then the other proteins will start attaching. So inner layer, outer layer, and that is how the sac would be formed, and then it is taken forward. So then these coverings are removed finally, and then again recognition takes place. So those complexes, just briefly I told about you, that clathrin coated vesicle. So that vesicle which is coated with clathrin. So it is, these are the central players in receptor mediated endocytosis. So they also mediate transport of cargo from trans-Golgi network to endosomal lysosome compartment. So this was the first. The other major players in this are adapter proteins, we call APs. So these APs are starting from one by one they were discovered. So they have been named as AP1, AP2, AP3, AP4 and AP5. They have been identified, that still we will get more. And these are very complex structures. These are complexes. This is not one protein. So AP1 is not one protein. It is a very complex protein, and it is just part, it is a one player out of these three main players which I told you in this transport mechanism. Now we'll have a look at the AP protein. So what we did is that my students, so one student did PhD in one AP, another did on another AP, another did on another AP. So this work was enough for one PhD, and we couldn't. We started from very high impact journals, so our papers were rejected, and we came down, and then this corona problem came, and I lost touch with my students, so I have to pick up the threads again. I have to start all over again. Around 2007, this first publication in Plants came about the intercellular vesicular trafficking in plants. So I thought that I'll be one of the main one. So we thought of designing. So just look at this. AP1 is composed of two large. So there are two large proteins in this present. That is gamma and beta 1. There is a one medium. So these are again group of protein. That medium we call mu1, and there is a one small sigma 1 subunit. So this AP1 has got the various subunits. So that subunit is made up of those. So each has got a specific sequence. Then we study all their sequences. Then we compare all those sequences with humans, with bacteria, with other like that. So AP2 is composed of two large. This is alpha and beta 2. You are familiar with hemoglobin protein. So it's much more complex than that. Then one medium mu2, then one small mu2 then there is AP3 composed of two large, this delta
Thank you very much, sir. It was a very informative lecture. Now, I would request Dr. Priyanka Srivastava to please present the
Good evening, everyone. First of all, I would like to thanks to all the organizing members who give me a golden opportunity to presenting here. I'm Dr. Priyanka Srivastava and I'm a postdoctoral fellow at CSA Agriculture University, Kanpur by ICSSR. Today, I would like to throw light on very serious, sensitive and progressive issue which is often overlooked. You know very well. My topic is impact of social media on body image and food choices in youth. Social media content is poorly regulated and targeted, as you all know very well about it. Food and beverages organizations are known to exploit young adults using image-based marketing tactics, including influencers and celebrity endorsement designed to sell on illusion of health. Celebrity evidence is now emerging of the negative consequences of such content for eating habits, health and body image concern, particularly in youth. What is the effect of social media on health and body image? The use of social media is generally associated with increased acts of skipping breakfast and consuming junk food and energy drinks. Second is children who have higher online engagement with food brands and content are more likely to consume unhealthy food and drinks. Research is increasingly showing that social media does indeed contribute to negative BI, and exposure to social media increases body dissatisfaction and disorder eating. People with negative BI, that is body dissatisfaction, feel dissatisfied with their appearance and perceive a discrepancy between their current appearance and ideal appearance. Let's talk about the numbers. Over 90% of adolescents have at least one social media account, as you all know very well about. 81% use more than one social networking site. 60%, 64% use social media 2 hours per day. 48% take selfies at least once per fortnight. 62% edit photos. 90% Facebook user check 3 to 5 times daily. 57% check Instagram 3 to 5 times per day. 22% reported seeing their favorite food brands advertising on social media. 6% reported hashtagging food and beverages brands on brands like Domino's, Pizza Hut, Burger King, you know very well, on any social media. And next is majority, 81.5% of teens in the US reported daily use of social media. Students who engaged in unhealthy eating behaviors were more likely to use social media at higher levels than students who refrained from unhealthy eating behaviors. Students who reported using social media for less than 1 to and more than 5 hours per day were at 1.67, 1.90 and 3.29 times the odds of consuming sugar sweetened beverages and energy drinks respectively. What's the pointers from review? Time spent on social media was inversely associated with self-esteem and directly associated with social comparison, depression, anxiety, and body shame. Selfie posting negatively correlated with body satisfaction. Friends fitness post negatively associated with body satisfaction. Appearance comparison and online fat talk directly associated with disorder eating behaviors. And next is association between social media engagement and BI and food choices outcomes were confirmed in 61% of the studies. What message do teens receive? You can see all these photographs. These are some type of ads which we get to see over the social media. You can see facts and stats. 95% of people with eating disorders are between the age of 12 to 25. Between 5 and 10 million teens in US currently suffer from eating disorders, and approximately 91% of females are unhappy with their bodies and resort to dieting to achieve their ideal body shape. So we conclude, research highlights the need to include regulations on social media and policies designed to limit children's exposure to unhealthy food, marketing, campaign ad brands like Burger King, KFC, Domino's, etc. Therefore, social media campaigns have a great role to play in protecting children from advertising. Social media engagement and exposure to image-related content have a negative impact on BI and food choice in healthy young adults. Population group who are vulnerable to social media influence. The pressure young adults feel to present in ideal image of themselves alters their food behavior and health eventually. How can we empower our teens? Firstly, health professionals should reach and engage with young adults to encourage healthy behaviors. Second is social media campaign must follow strict guidelines so that it doesn't unintentionally create or promote further body dissatisfaction among young adults. And next is reducing youth's screen-related sedentary time and increasing our effort with message on healthy food choices are important public health target to achieve. And next is focus on a positive relationship with food and exercise more. Thank you so much. When you change the conversation with yourself, you will change the conversation with others. Because according to me, your body is your temple. So it need to keep it neat and clean for the soul residing. Thank you so much.
Thank you, Dr. Purification, for your diversified presentation in the social science related. And I think all the audience will benefit by your this presentation. Thank you very much. If any question from audience, then they can ask.
I just heard your last part of it. You are worried about yourself, but you are polluting the environment of animals.
Sir, please, Mike.
You are worried about yourself.
No, sir, I'm not talking about myself only.
Myself is human.
Okay, yeah, yes, sir.
But what about others? You are showing your letters to the this jungles and all there, these all animals they used to live and eat from the ground. And they are littering this everything for them. So first, it is not we.
Yes.
If we have the environment clean for every living creature on this earth, then only we can survive. If I get sick, I am worried.
Yes.
But if my animals are dying, other thing there's so much loss of diversity,
yes,
lost in insect population, lost in plant varieties, so and so forth. तो अकेला आदमी रह क्या करेगा?
Sir, because my topic
The
is
social means it involves everything. It's not comprehensive. It's not one human being.
Okay, sir.
Human being is a very small part of
Yeah.
the entire
Okay.
ecosystem.
Okay, sir.
So think of that.
Okay, sir. Thank you so much.
Nice suggestion by Dr. Producing sir. I think any more question from audience side?
There's a person.
मेरा जो क्वेश्चन है वो बिल्कुल जनरल है भाई, मतलब ऐसा होता है कि जो सोशल मीडिया है उससे डिस्ट्रैक्शन बहुत सारा है, लेकिन क्या होता है कि जब हम उससे दूर होना भी चाहते हैं तो ना चाहते हुए भी हम उसके पास जी मैम, तो क्या होता है कि हमें जब हम कोई भी सोल्यूशन भी ढूंढना चाहते हैं लेकिन वो हमें सोल्यूशन नहीं मिलता है, लेकिन मैम व्हाट इज परमानेंट सोल्यूशन ये चाहता हूँ।
सो परमानेंट सॉल्यूशन तो किसी भी चीज का नहीं है, लेकिन थोड़ा सा विल पावर स्ट्रॉन्ग करने की बात है। अगर इंसान थोड़ा सा विल पावर स्ट्रॉन्ग करे तो कुछ भी संभव है। एनीथिंग इज पॉसिबल इन दिस वर्ल्ड इफ यू वांट टू डू समथिंग। सो मेरे ख्याल से तो आप अपने पर रोक खुद लगा सकते हैं, कोई और नहीं लगा सकता, और उसके लिए सबसे इम्पोर्टेंट चीज अकॉर्डिंग टू मी विल पावर।
क्या करें, पूरी यूनिवर्सिटी में उस पे चलती है।
Sir, benefits is other thing. I'm not talking about.
वो तो मेरे को बोलते तुमने देखा नहीं मीटिंग थी, बोले इसको कितनी बार देखो।
लास्ट 2 से तो पूरा मतलब
बिल्कुल
चाय भी नहीं छोड़ सकती हम।
बिल्कुल छोड़ने के लिए सर मैंने बोला भी नहीं, इसमें ये बिल्कुल नहीं है कि आप इसको छोड़ दें, बट एडिक्शन नहीं होना चाहिए सर। बच्चों के लिए, आपके लिए जरूरी है सर, बच्चों के लिए ऑनलाइन क्लास के लिए भी जरूरी है, लेकिन इसका ये मतलब नहीं है सर उसका बहुत ज्यादा ऐसा यूटिलाइज होने लगा है कि बच्चे कितने ज्यादा
ग्राउंड नहीं छोड़े।
नहीं, कोई नहीं छोड़ सकता। मैं सर खुद इस पे लेक्चर दे रही हूँ, लेकिन मैं नहीं छोड़ सकता सर। बट आई एम टॉकिंग अबाउट द साइड इफेक्ट्स ऑफ दिस। मैं इसके साइड इफेक्ट की बात कर रही हूँ। ऐसे हर चीज के बेनिफिट्स होते हैं, कुछ लॉस होते हैं, तो मैं यहाँ पे सिर्फ इसके कुछ जो लॉस है मैं उसकी भी बात कर रही हूँ। तो इसके लिए बस मैं ये कहना चाहती हूँ कि जो सोशल मीडिया है उसकी वजह से हमारे बच्चे, हम लोग अपने पे तो कंट्रोल कर भी सकते हैं विल पावर स्ट्रॉन्ग करके, बट बच्चों को समझाना बताना बहुत मुश्किल होता है। जब बच्चे जिद पे अड़ जाते हैं कि मम्मा ये टीवी पे ऐड आया मुझे ये चाहिए, और लोगों के लिए थोड़ा सेल्फ एस्टीम वाली बात भी हो जाती है, तो कहीं ना कहीं हमें ना चाहते हुए भी करना पड़ता है। मुझे जंक फूड बिल्कुल मैं उसके अगेंस्ट हूँ, बट मुझे अपने बच्चों को कभी महीने में एक बार इसलिए खिलाना पड़ता है, फ्रैंकली स्पीकिंग, कि कहीं कल को उसके फ्रेंड सर्कल में बात हो तो ऐसी बात ना आए सामने कि बच्चे को तो मेरे पता ही नहीं है, तो वो मतलब ऐसा फील करे कि मेरी मम्मा ने तो मुझे कुछ बताया ही नहीं। तो मैं उसको महीने में एक दिन मैं जंक फूड देती हूँ, सिर्फ इसलिए कि सोशली उसको कहीं बुरा फील ना हो, एम्बैरस ना होना पड़े, इसलिए सर कहीं कहीं कंपल्शन है, बट मैं छोड़ने की बात नहीं कर रही हूँ, बट थोड़ा सा विल पावर स्ट्रॉन्ग करके इसको थोड़ा कम कर किया जाए, क्योंकि ये कहीं ना कहीं बहुत हार्मफुल है सभी के लिए।
Thank you very much, ma'am. Now it's already 6:17 and we have to conclude with today's program. The cultural programs have been postponed for the last day due to some reasons. We'll be now request the chairperson of this session to please give his kind review of this session.
Good evening. In this session, we first go through Dr. I.S.G.B.R.D of SNN College, Delhi University, on the topic viral vaccination. He gave very concise, whole information about what happened in the pandemic, and what are the issues among globally, and what are the doing by the scientists for the vaccination purpose. After that, we find a very nice presentation by Dr. Survi Singh sir, and earlier he is a scientist in this Furry Institute of the Central Institute of God, and on the livestock, and I really appreciate his work because he just invented or discovered this vaccine for John's disease and TB also. Then after the presentation from by Dr. Pankaj Gupta ji and Ravi Upadhyay ji gave all about diversity of the Panchmali, short presentation by Dr. Ravi, and then after Pankaj Gupta, अनुभूति का बाद में हुआ था, तो पंकज गुप्ता जी just give, I think, a recent branch of this biosciences, I think, work in biotherapeutic and biomedical engineering related matter. Being the plant scientist, we are here in the committee of this session. I think that was very beneficial for those who is working in advanced biosciences. And then after Anubhuti Sharma ji gave very nice work. I think this is a good work for this mustard, and he gave information about what are the various issues of edible oil and what are the research is doing by the ICR and this year's Bharatpur Institute in the research of mustard. And then we have Dr. Randhawa sir give good account molecular biology or recent work. I have never listened this transport of the protein. I think youngsters, even the bee people of who are working in plant sciences, get some idea of innovative lecture. I really find it is very good work done by Dr. Randhawa, and all the audience will get benefited. And the last topic by the Priyanka Srivastava ji changed the session in this social science related. The social media impact on the youngsters and even all the human being scientists, as Dr. Sonvi sir said कि हम लोग का जो पेंडेमिक में सारा इंफॉर्मेशन ईमेल, जो लोग लिटरली नहीं थे, हम लोग कंप्यूटर से बहुत ज्यादा नहीं रखते थे, आज सोशल मीडिया से बहुत सारी इंफॉर्मेशन मिल रही है. बट जो मैडम का इम्पैक्ट था, उनका जो प्रेजेंटेशन था, दैट इज फॉर द यंगस्टर कि सोशल मीडिया की जो नेगेटिव चीजें हैं, और मैंने अपने घर में भी फील करा कि जो बच्चे हैं उनका कंसंट्रेशन कम हो रहा है, और जो उनकी लाइफ स्टाइल है, उनका जो हैबिटेट फूड है. मैं बताऊं आप लोग को कि मैं एमएससी कर रहा था तब मैंने फर्स्ट टाइम चाउमीन, मतलब मुझे पता चला कि चाउमीन सीएसए में मेरे एक फ्रेंड के पास में रुका तो पता चला. तो ये इम्पैक्ट है, और ये बहुत अच्छा प्रेजेंटेशन था, शॉर्ट टाइम में इंफॉर्मेटिव है कि हम लोग अपने ई टाइम को कम करें. सो आई थिंक दिस सेशन whereas I think very benefited for all those concerned online and offline in this biosciences, and I just give rest to my words, and I appreciate the organizer for this wonderful congress. Thank you very much.
Thank you very much, sir. Now I would request Dr. P.K. Upadhyay sir to please come forward and honor our invited guest speaker, Honorable Professor G.S. Randhawa sir from IIT Roorkee, with shawl, patka and the bag which we are giving, and memento. Professor G.S. Randhawa sir, a huge round of applause. Now I would request Dr. Anurag Bajpai sir to please honor chairperson of this technical session, Professor B.K. Chaturvedi sir, Head Department of Horticulture, Agra University. I would also request Dr. Arun Chopra sir to please come forward and honor our co-chairperson for the session, Dr. D.S. Chopra sir, Department of Agronomy, RBS College, Dr. B.R. Ambedkar University, Agra. Next in the sequence, I would request Dr. Ajay Kumar Sharma sir to please honor the reporter for this technical session, Dr. Manoj Pandey sir, with the Patka cloth and token of gratitude. Tomorrow in the evening, we are having a special cultural program. We have invited a rock band from Hindustan College of Science and Technology, which will be presenting a few songs and whatsoever they do. Also, we have planned a tour excursion to Mathura and Vrindavan in the morning. The bus will be leaving from Hindustan College of Science and Technology, Parel, at 6 a.m. sharp in the morning. The breakfast packets will be distributed in the bus only. The technical session 5th will start tomorrow sharp at 11:30 or 11:10 as per schedule. Thank you.